Good afternoon, everyone. Let's get started. We have anxiously, excitedly waited for this day to have the spatial symposium. Hereby, I'm Nidhi Mohan, Sales Development Representative here at BioChain Institute. It's my pleasure to welcome you all to our third annual spatial symposium here at the beautiful lighthouse in South San Francisco. Heartily welcome to everyone who are present here as well as online. You know, spatial has been quite a labor of love for all of us over the last few years. As this technology was emerging 4 years ago, we recognized how transformative it could be and how closely complex multiomics could align with BioChain's existing histopathology and NGS capabilities. So we chose to take that headfirst dive, and it has been this breakneck journey of incredible learnings and even more incredible partnerships ever since. As we do every year, we have gathered leading experts to share cutting-edge advances in spatial biology, an area where BioChain has been investing deeply to make spatial workflows simpler and more accessible. Over the next 3 hours, we'll dive into the first 5 transformative areas of spatial biology. Number 1 being Visium HD multimodal mapping, where we expand RNA and protein detection on a single slide for richer tissue insights. 2 being the precision validation on 10x genomics Xenium and how BioChain's rigorous testing ensures data you can trust. Third, deep multi-omic context through leveraging 2D and 3D image data to unlock true cellular complexity. Fourth, multiplexed TSA fluorophore assays, the simultaneous detection of proteins, RNA, and glycans in workflow. And fifth, but not the least, high-throughput FFPE tissue profiling via singular genomics, how to bring integrated single-cell multiomics to your archival samples. Before we begin, we know spatial is complicated, and here is why at BioChain we make it simple. Today's presentations will not only showcase the latest technologies, but also highlight practical tips and best practices to streamline your own spatial projects. Before we dive in, a quick housekeeping note to all our in-person attendees that you have been automatically entered into a raffle to win one of the 3 exclusive discounts on their first spatial project with BioChain. So without further ado, it's my honor and privilege to introduce the new CEO of Cell Bioscientific, Charles Miller, who shares our excitement for BioChain's next chapter. Miller holds a BS and PhD in biomedical sciences from the University of Michigan and Michigan State University, respectively, and an MBA from Yale. His management style is characterized by clarity, resilience, and a commitment to building high-performance cultures that deliver measurable impact. With more than 15 years of leadership experience in drug discovery, diagnostics, and preclinical services, Miller brings a proven track record of transforming organizations and delivering sustainable growth. Miller brings a proven track record of transforming organizations with his previous roles at the Jackson Laboratory and Eliphas Biosciences, including leading global commercial operations with revenues exceeding 500 million, brand building in life science and drug discovery, and driving market expansion into more than 70 countries. Please join me in welcoming Charlie. Lead the way, Charlie. Give him a big round of applause.
Wow. Well, thank you for that wonderful introduction. Good afternoon, everybody, both participating here in person as well as online. It's my pleasure to welcome all of you to our third annual spatial symposium hosted by BioChain, which is a cell bioscientific company. As I was introduced, my name is Charlie Miller. I'm the CEO of Cell Bioscientific. And I think I wanted to thank all of you, whether you're a researcher, a partner, or just simply curious about where the field is headed, we're all looking forward to a wonderful program today. As you're all keenly aware, we're at a pivotal moment in biomedical research where excuse me, where the convergence of spatial biology, single cell analysis, and advanced imaging technologies is really redefining how we look at biomarkers and biology in cells and tissue biospecimens. And so we're at a point where we're harnessing those technologies to see what advances we can make, not only in basic research, drug discovery, but also harness these technologies for clinical application and potentially intervention as well. At Cell Bioscientific, we're really proud to be a platform company that's built through the integration of three pioneering organizations, BioChain, obviously, as well as System Biosciences and AcePix Biosciences. Together, we offer an end-to-end portfolio that spans biospecimens and clinical tissue, spatial biology, as well as biomarker analysis, cell-based models, extracellular vesicle technologies, as well as gene delivery tools and services, serving both preclinical drug discovery markets as well as clinical histopathology services, as well as products. And our mission is simple. It's basically to empower drug discovery scientists and clinicians with the tools and services that are necessary to advance the next frontiers in human health. And so just briefly introducing some of our specific companies and platforms, AcePix, which was founded in 2017 in San Jose, California, from founders that were principal assay developers at ACD. We house the world's largest portfolio of FFPE immortalized cell pellets that are unique next generation tissue surrogates that are used by biomedical researchers, drug discovery scientists, as well as use in clinical diagnostics. We offer full service histopathology services and are a global leader in tissue embedding, preservation, preparation, and digital imaging analysis. System Biosciences was founded in 2004 in Palo Alto. It's been a pioneer in extracellular vesicle isolation and purification since our founding, and a global leader in gene delivery reagents and services. And we also have an extremely active R&D program focused on pushing the frontiers in both of those spaces. And obviously, you're all familiar with BioChain, which was recently acquired by Cell Bioscientific in May of this year, so last month. So we're excited to add BioChain and its core services and biospecimens to the expanding portfolio at Cell Bioscientific. And so why host a symposium focused on spatial biology? And for us, because it really sits at the field sits at the crossroads of really everything that we do. It's one of the most promising and rapidly evolving areas, as all of you know, and it bridges pathology, genomics, as well as histopathology. And spatial technologies are helping us move beyond traditional approaches and allowing our customers to ask not just what is expressed, but how gene expression influences patterns of biology within the organizational architecture of tissues and the microenvironment. And so it's that change in resolution in terms of the biology that's allowing us to better understand cancer, neurodegeneration, inflammation, and countless other diseases so that we may potentially diagnose and intervene on those diseases more effectively. And so combining the three companies within our portfolio, we're expanding. We've put together and we're expanding a completely integrated cell and tissue analysis platform across Cell Bioscientific, within which spatial biology makes up an important component. And so we provide human as well as animal model tissues and cells, biospecimens within our repository. We also do custom sourcing of those. We have an expanding line of cell models as well as tissue models. and the capabilities to engineer cells and tissues for our customers in both the drug discovery and basic research space. I talked a little bit about our complete histopathology workflow and downstream services, including assay services like advanced staining and IHC and molecular isolation as well. And then furthermore, the deep spatial analysis, as well as digital scanning, capture and analysis of tissues, and pathologist review as required. And so, we're not just spectators in this space. We're really focused on supporting our biopharma clients through spatial biomarker discovery, working with the innovators and the technology providers in the space to provide the best services possible that we can to our customers. We provide IRB consented tissues and fresh frozen samples, as well as we're partnering to create completely integrated workflows that combine spatial omics, AI-powered analytics, as well as machine reliable pathology. And so today it's not just obviously about Cell Bioscientific, but it's really about the spatial community and working together to advance the science. So you'll hear from pioneers in the field. You'll learn about some of the breakthroughs, bottlenecks, challenges, and innovations that are happening across the space. And our goal with this symposium, whether you're participating here in person or online, is to spark a dialogue, to create a space where the technical expertise can meet some of the scientific innovation that's happening in the field, and where meaningful collaborations can take place. So again, I wanted to thank all of you for participating here in person as well as online. Thank all of our participants and speakers for being here as well, and looking forward to kicking off the substantive portion of the conversation. And so with that, Peter, I'd invite you up here.
Thank you, Charlie, for your introduction to Cell Bioscientific as a fully integrated platform for cell and tissue analysis. It definitely offered a clear view of where we are headed to. We also appreciate your emphasis on our growing role within spatial ecosystem. It's exciting to be a part of a journey that's shaping the future of biomarker discovery. And next, moving up, I want to really kick off our technical sessions by inviting Dr. Peter Smibert, Vice President of Biology from valued partner 10x Genomics. Dr. Smibert has a rich doctoral background in genetics and molecular biology, where he leads teams spanning molecular biology, cell biology, and spatial applications. Prior to his work at 10x, Dr. Smibert served as a director at the New York Genome Center. Dr. Smibert has been instrumental in advancing single-cell and spatial biology techniques. He and his team have been pioneers in expanding Visium HD to new species and integrating RNA and protein detection on a single slide. Please join me in welcoming Peter, who will be presenting on the topic Visium HD and Xenium RNA plus protein detection. Over to you, Peter.
Thank you. Sorry, you're getting a very brief sneak preview. We ended up at the thank you slide. So, all right, any questions? Hi, thanks for having me. It's really nice to be here, and interacted a lot with BioChain over the years, and it's nice to come and present here. So thank you for the invitation. So I'm just going to speak on behalf of 10x Genomics R&D. There's a huge number of people that do this work. I'm just the one who's lucky enough to come and present it to you all. This is a spatial symposium, but it would be remiss of me not to just spend a couple of slides talking about Chromium. This is 10x Genomics. We've cut our teeth in single-cell genomics and expanded into spatial genomics. But I think there's things that we do, the way that we approach our single-cell business is also the way that we're approaching our spatial business. We really want to emphasize quality. We have a broad menu of different assays, analytes, applications. Performance is super important to us, as is customer experience. And I think we're doing some estimates about the number of cells that have been profiled, and less of an estimate, but more an exact number of the number of publications that have been used, come out with our techniques over the last 10 years since we've had these products. The things I do want to emphasize, as I mentioned, we really do place an emphasis on performance and also value. And just a couple of little snippets here. On the left, it's a preprint from the Fred Hutch that came out recently that's done a bake-off of a bunch of commercial single-cell products. Some most current, so our GemX products are the most current. Next Gem is our older generation. And I think we're very pleased with how these came out in terms of both showing off our sensitivity on the left-hand side, and then from an independent preprint on the right-hand side that actually shows the read utilization. So you think about how many of your reads that are generated in your assay actually go towards producing usable data as opposed to being wasted data or not in cells. And I think that the data speaks for itself, that is independent of us, that we really want to emphasize that we both excel in quality, but also the value that we bring to you, because you're not wasting a bunch of money sequencing garbage that you don't need. The last slide I'm going to have on single cell, I promise. You may have heard about our Flex product. This is a method to be able to detect, to do single cell RNA sequencing on fixed cells, up to and including FFPE samples. You can dissociate FFPE scrolls and run single cell on them. We're drastically expanding the scope of this product to the point where we're going to have 96 and 384 well barcode combinations for this product, allowing you to load at least 1 million or recover at least 1 million cells per chromium lane. So shown here, I don't know if you can see my cursor. Is that visible? No? Anyway, one lane of a GemX chip can recover more than 1 million cells. And we're super excited about this product because we think it's really going to open up a lot of applications in things like screening, but also just flexible utility. You can run 5, you can run 48, you can run 96 in a way that you fully utilize the kit and there's no wasted reagents. So we're not locking you into experimental configurations that you don't want to use in order to get the best economics. You can choose how to get the best economics out of this product. All right, I promised to be short on single cell. That's it. That's all I'm going to say about single cell. The other part of our business is looking at spatial biology, and we have 2 offerings in spatial biology that were mentioned in the introduction, both offered by BioChain. There's our Visium method for spatial transcriptomics, where we essentially capture the analytes from a tissue onto an arrayed slide and report the spatial localization. And then there's Xenium, where we actually do in situ sequencing within your sample, generate molecules that we can read out and get the precise location of individual transcripts and proteins within that sample. So I'm going to walk through these products in the next little while. So starting with Visium, this is our method for whole transcriptome spatial discovery. As with everything that we do, we don't just produce an assay and give you data and wish you good luck. We have instrumentation that makes your life easier, makes the work more reproducible. We obviously have the assay, and we have the downstream software and analysis that helps you get from sample to answer more quickly, so that you're not stuck in this paralysis position where you have a bunch of data and you're not sure what to do with. We can really help you analyze, visualize, and interpret your data. We have a couple of different versatile HD assays to meet your needs. On the left-hand side, the current product that is on market and for sale right now is the probe-based whole transcriptome panel gene expression. We use a chemistry here with probes. So again, this is designed to go where the samples are. It's not, we would love it if we had complete control over every sample that you guys get for your research. The fact is you don't. A lot of the time you're starting with blocks that have been processed by someone else at some distant point in time, preserved in FFPE, and that's your starting point, and there's nothing you can do about it. So we've designed assays to go after that sample type. We can make this probe-based assay that can really effectively measure transcripts and proteins in FFPE samples. On the right-hand side, this is our Visium HD 3' gene expression. For those that have run Visium in the past, this is how we initially did the Visium product, where you're, instead of capturing probes, you're capturing mRNA that is released from the cell onto the array and measuring its location in spatial proximity to where it was originally located in the tissue. And there's obviously reasons why you would want to do this. You're not limited by the species of origin or the species by which the probes are designed against. You can also do things like de novo discovery. You don't necessarily know what you're going to measure, but whatever you pull down and capture on that array, that's what you're measuring. So you can go after things like isoforms, TCR, BCR, and much more. I mentioned our initial Visium HD with our probe-based chemistry. So this is kind of how it works. I tend to just do it with my fingers here. You have a left-hand side probe and a right-hand side probe. they sit down on top of a transcript of interest, and only when they sit together and align perfectly are they ligated together. And so that ligation together tells you that transcript was there. And then we barcode that probe. So the ligated probe gets barcoded and captured and read out on a high throughput sequencer. And right now we have probe sets covering effectively the whole transcriptome of both human and mouse. Our single cell resolution is really enabled by our high definition slide architecture. So we've made really significant strides here. I don't have a comparison here against the older version of this, but the older version had 50 micron spots. So let's say about that big. And they had 100 micrometer pitch in between them. So there's a bunch of empty space in between. Our newer version has 2 micron spots that has no gap in between them whatsoever. So 2 micron squares. So it's a continuous lawn of oligonucleotides. We've gone from having something like 5,000 individual barcodes on a 6.5 by 6.5 millimeter area to more than 11 million barcodes. So it's really a significant increase in the resolution of the array. But, and I'll caution, the resolution of the array is not everything in a spatial assay. And the reason is molecules move. If you're moving molecules from your sample to a slide and you want them in as close a proximity to where they actually originated as possible, you need to be able to control that diffusion. So we initially started the Visium assay as a direct placement assay where you would take a slice of tissue and lay it on top of the array, lyse the tissue, release the contents, and off they would go, and you'd hope for the best. We have since developed this CytAssist instrument. And this is very simple, the way this works. You have your samples on a slide, you can do your standard histology and imaging, you have your barcoding slide in the middle, and this instrument essentially sandwiches them together. This instrument, the code name for it during development was actually Foreman, because it kind of resembles a George Foreman grill. Doesn't really cook things the same way, but you get the idea. But this is sort of a schematic as to what this looks like and why controlled capture is important. So on the left, this is what happens with CytAssist. You have your sample is bounded by the slide on top, and your array is bounded by the slide in the middle. We bring them together at a very controlled distance apart with controlled buffer conditions and stimulate the transfer of the analytes from the sample to the array. When we do direct placement, it's a much more uncontrolled capture. You're putting the sample on top, and then you're applying a buffer to the top of that sample to start lysing it to release the contents onto the array. And as you can just sort of empirically see here, things are going to move every which way. You're going to get some correct localization, you're going to get a bunch of mislocalization. So this is the end result. This is what it looks like when you actually combine a very high definition array with a very precise controlled means of transferring the analytes from one side to the other. And these are just some examples that we actually use in development where we would find highly expressing genes in cell types that are near each other, but they're mutually exclusive in their expression pattern. And this is essentially how we would say, is our assay working? Do we have good spatiality of our data by going and looking at these pairs of transcripts? And we have obviously run this on a bunch of different samples now. This is a FFPE on the left-hand side, fresh frozen or fixed frozen across both human and mouse conditions shown here. We can also generate exceptionally rich data. So while we have designed this initially with FFPE in mind, you can take fresh tissues and convert them. You can fix them in a way that makes them compatible with this assay. And when you do this, you actually get incredibly high sensitivity of transcript detection. And this is actually shown sequencing with ultra-genomics, which is an interesting approach here because now the sequencing becomes a lot cheaper and you can generate a lot more sequencing reads. And so you can take these really sensitive, the sensitive assay with high expressing tissues, sequence to saturation, get a much clearer view of the biology within your tissue. In development, we are increasing the size of the capture array. So at the moment, this is all just schematized here, but 6.5 millimeters is the small little square here. 11 millimeters is the large square here. And this is just a couple of examples of how much more biology you can capture once you have a larger imaging, a larger array here. But everything I've talked about so far is this probe-based chemistry where you know what you're measuring because you've designed probes to it, and that's precisely what you're measuring. It's the presence or absence of a ligated probe pair indicating a transcript was there. But people have been pushing the boundaries of single-cell biology and also spatial biology, trying to use, these are RT-based methods where you don't necessarily know what you're going to capture. And there's reasons why you want to do this. You might want to profile a wide variety of different species. We've got here chicken, pig kidneys, xenograft, rat brain, or you want to look at applications beyond just transcript counting. You might want to look at splice variants, profiling isoforms, looking at B cell and T cell receptors. So these were some of the reasons why we were motivated to extend Visium HD into an RT-based chemistry as well. And I think I covered some of the reasons here, but this also works very closely with CytoAssist for the same reasons that I mentioned in the past about really wanting precise analyte transfer between the tissue and the slide. And so similar slide that I showed before, it's actually the same slides that we use. They're treated slightly differently. The workflow is a little bit different, but now instead of the ligated probes transferring across, we now have the mRNAs from the cell transferring across. And this works really well. We've designed this nicely to be compatible across species with no real optimization. Human and mouse tissues, of course, which a large proportion of our users use, but then also on the right-hand side, a variety of different species that are all compatible with this approach. I think I put my slides a little bit out of order, but this is just to show you nicely what the difference is between the 55 micron spots that we had in the previous version of Visium to Visium HD. And these are adjacent sections of a fresh frozen mouse spleen. So we're down to single cell scale now. And with CytoAssist enablement, we get much better spatiality and a really universal single protocol over a wide variety of different tissues. The sensitivity, frankly, we wouldn't release the protocol unless it was better than the previous version. In blue here is Visium HD 3' compared to Visium V1 in red. And you can see that in all conditions, it either meets or exceeds the sensitivity of the previous version. And I think this is just a nice demonstration here as to why you might want to use this if you're deciding. It depends on the research that you're doing, but why you might want to go with a probe-based assay or go with the reverse transcription based method. You still see a lot of the genes in common. So this is genes actually detected in the experiment. There are more genes, obviously, but not every gene is going to be expressed in a mouse heart. But you can see you get unique genes from each chemistry in this particular example of adjacent sections. As I mentioned, you can explore any species with an annotated transcriptome with this method here, and this is just an example of a kidney from a rhesus macaque. You can see really fine-grained structure here, and you look at individual marker genes for distal tubule cells, proximal tubule cells, or immune cells within the kidney structure. And this is just another example as to why you might want to use an RT-based assay for Visium, where you have a xenograft here, and you can very clearly distinguish the human tissue from the mouse tissue, depending on the question, if you want to look at the human tissue or the mouse tissue, or the interplay between the two. I do want to just spend a minute now talking about how we're expanding the power of Visium HD with cell segmentation. So I talked about that we have these 2 micron squares that we use for barcodes. Initially, we had lumped these together into 8 micron squares, 8 micron bins. So 4 by 4 of these 2 micron squares. This is essentially this is a lot of barcodes, and the data, if you go 2 by 2, can be fairly sparse, as you would expect over such a large number of barcodes. But what we've now moved to is taking advantage of the fact that this instrument, the CytoAssist instrument, has a built-in camera. It takes nice images it takes nice H&E images of the tissue. We can use the H&E stain to define nuclei and use that to segment the cells. And so now we can assign these 2 micron squares to cells, either based on just the nucleus, as shown sort of in the middle right here, or if we expand these bins out into other cells, or being a bit more greedy and taking a little bit more space around the nucleus on the right-hand side. And if we do this is just an example here from an FFPE human colon, just the H&E image shown on the right. In the middle, you can see if you zoom in here, we actually have our software allows you to annotate the nuclei and then do segmentation-based clustering here. And so this is built into our Space Ranger software now, straightforward pipeline, and it's integrated into Loop Browser version 9. And this is a reason why you would do this. So here's just an example here from a human tonsil, what it looks like if you cluster just by 8 micron bins in the middle here. And if we zoom into this sort of nondescript section in the middle, that does look a little bit nondescript with the 8 micron clustering, but we do segmentation-based clustering instead. you can actually see, you can start to discriminate different cell types. So there's different flavors of B cells in this region that are defined differently by their transcriptome that you can only detect if you actually take the nucleus and make sure you're assigning the correct transcripts to that individual cell. So just to summarize the Visium section that I've spoken about today, we have two different flavors of Visium HD. There's a whole transcriptome probe assay that's really suitable for FFPE, fresh frozen or fixed frozen compatibilities. We also have a 3' assay that is super useful for fresh tissues. And this would be the application if you want to move beyond human and mouse, if you have access to nice fresh tissues, and you really want to, you don't know what you want to measure, you want to see what's there. I also mentioned that larger capture area is on the horizon, and the cell segmentation analysis is now integral to both of these assays, and is something that can work through our software. Okay, I'm going to move into Xenium in situ now. This is the sort of the other half of our spatial offering. I think I love this GIF because it just sort of shows nicely what Xenium can provide, and it's oscillating between, let me see where I can start, between the H&E image here, moving to the cell segmentation stains, what the clustering looks like on top, and then just all the transcripts that are actually detected in that particular section. I showed this to my daughter and she sort of said, "Oh, Dad, it looks like one of these." So, Xenium's been on the market for a bit over 2 years now, and it's great because it's been in a lot of people's hands. People have a lot of options out there in terms of spatial biology and things that they can measure, especially for in situ methods. Any vendor will get up and tell you their product is great. I like to just be able to point to the fact that there's a whole bunch of different bake-off studies out there that compare our products to some other products in the market and consistently show the same things, detecting more transcripts on shared genes, discriminating cell types much better, showing much better specificity, and really being incredibly consistent with orthogonal benchmarking such as single cell assays. So I encourage, if you're considering different spatial assays, to don't take our word for it or anyone's word for it, but read the independent papers. So Xenium is a panel-based method. We have a broad panel menu that really can suit a bunch of different applications. Our V1, we started out with tissue-specific and application-specific panels, both in human and mouse. This includes multi-tissue, immuno-oncology, brain, breast, lung, colon, skin. And then in mouse, we have multi-tissue and brain. We've also released a 5,000 plex panel that's on actually quite a different chemistry in the way that's, I'll go into this in a little bit, on both mouse and human. And customers can add up to 100 custom markers to any one of these panels. We also have a bunch of customers that are taking this and making fully custom panels. So if they're working on a completely different species, or they work on a tissue that our targeted panels don't measure, or frankly, they say, we don't like your breast panel, we prefer to make our own, that's totally fine. And we can enable that with our applied bioinformatics team. There's also advanced customization, which I'll speak about in just a second, where you can go after additional sequences here. The beauty of the way the Xenium chemistry works is incredibly specific, even down to the nucleotides. So you can detect things like splice variants, single nucleotide variants, and things like TCR, BCR profiling. And of course, it would be remiss of me not to mention this is again designed with the concept in mind that we can't control the samples that you guys get. So we started FFPE first. We wanted to make sure this was an assay that would work on FFPE and then work backwards from that. I think if you can get it to work on FFPE, you can always convert a fresh tissue into something that's more like FFPE. And so there's a wide variety of different sample types that Xenium can work over. So I mentioned Xenium Prime 5K. So this is our 5,000 plex panel that's available on both mouse and human. We've changed the chemistry actually pretty significantly. It doesn't feel like that from a customer's perspective. Your experience is not too different. But this allows us to increase the density significantly and increase the code book so we can encode a lot more transcripts. Importantly, we've maintained the high per gene sensitivity, and we've really kept the time down to make it practical that you can do this assay in less than 1 working week. So you can measure 2 slides at a time. We sort of use the 4 1 centimeter by 1 centimeter sections, so 4 centimeters squared in 6 days, which is a huge number of cells. This would be depending on the density of your sample, this could be a couple of million cells. This is just an example of what this looks like in a lung adenocarcinoma sample. This is showing clustering, some summary stats on the right-hand side. And if we zoom in here, just to give you an idea of what in situ sequencing actually means, just the precision of the localization of the transcripts, you can actually see macrophage markers in a macrophage that's the cell is identified and is sitting right next to T cells with T cell markers. On the left-hand side, and same sort of concept with Treg cells and fibroblast here on the right-hand side. I also mentioned just the sheer scale of this data. So this is a full well scan. This is essentially a 2 by 1 centimeter section of, I think this one's liver. It's been a while since I looked. Yes, human liver. There's more than 1 million cells in this one section here, and detecting a large number of transcripts per cell. This took 6 days to run 2 full slides. This is only one of the slides. So you can imagine the sort of scale of the data that you can get out from this. I mentioned Xenium Advanced Customization. This allows you to do very unique applications. I sort of did my finger demonstration of what flex probes looked like before. Well, now for Xenium, I can do the heart thing for my daughter again. Flip it around. Now you've got the parts that are binding to the transcript are my thumbs. It's a padlock probe. It's a circle. So what happens is the 2 arms of the padlock bind on the RNA. The junction gets ligated when the matching is perfect. And then we do a rolling circle amplification process to make a copy of something that we then detect subsequently downstream. Now, the reason I'm saying this is it's incredibly precise. These 2 arms, you can control exactly what they are. You can put it so there's a mismatch right at the junction location, in which case it will not ligate. You don't get a product. But if you think about you want to do a splice variant, you've got exon 1, exon 3 here. You can put them together and see if you get exon 2 being skipped. You can do these really precise measurements. And this is just a couple of examples of this. People going after exogenous sequences like viruses in human cervical cancer. I think the splicing isoform is a really nice example of this in the middle, or an SNV. And also, of course, I mentioned fully custom. You can go after non-human, non-mouse species, and we've had customers using tilapia or maize embryos. You can also use Xenium for analysis of cell suspensions. And so this is something that we've shown for a while and some other customers have shown back in 2024, but towards the end of the year, actually a big group of researchers came together and put out a method called STAMP. That's really cool to see. So they've figured out how to fix suspension cells, immobilize them onto a Xenium slide, and then just run our standard Xenium assays on them. And the number of cells you can get here are really scary. We're talking here, I think, on the left-hand side is the STAMP paper, and in the middle is an example of this from our internal R&D, where you can just plaster these cells on top of your slide to the point you've got 3 million cells in this one image here, and detect, for profiling a very large number of cells, it's an extremely powerful method. And of course, I think going forward, you can see people partitioning the slide in different ways, using different gaskets, etc. There's a lot of different applications that people are using this for. Okay, what I want to talk about in just the last bit of the slide is something new that's coming soon, which is doing RNA and protein on one slide with one workflow on Xenium. And here I just want to make it clear that we're focused on sample prep. We always try and make sure that we can make a sample preps as universal as possible, make it as hands-off as possible as well on the Xenium instrument. You press go on the instrument and the instrument does everything for you. And then there's integrated data visualization, of course, and analysis on the back end. So we will be releasing an initial panel of 28 different markers shown here. These are going to be different subpanels that you can choose to use all or some combination of. Protein customization is on the roadmap, where you'll be able to introduce your own markers that you're interested in into this. And this is going to be compatible with our tissue and application RNA panels. But why would you want protein and RNA at the same time? In my former life before 10x Genomics, when I was at the New York Genome Center, my group actually, if I fast forward even further back, I was a postdoc studying post-transcriptional gene regulation, looking at microRNAs, looking at other methods by which protein and RNA don't necessarily correlate. At the New York Genome Center, group of us had worked on similar problems in our postdocs and tried to apply the same thing to single cells and came up with this method called site-seq, where you take oligo-labeled antibodies and detect them at the same time that you're detecting transcripts. So this has always sort of been something that's near and dear to my heart. And it's very clear from doing this research that sometimes protein markers are just much better markers for cell types than transcripts. They're much more highly expressed. But then also sometimes the protein expression and the RNA expression just don't correlate. Post-transcriptional gene regulations are gene regulation mechanisms are so important for biology that it's really important to be able to measure these things. And going forward, looking into the future, you might want to be measuring different protein isoforms. You might want to be measuring phosphorylation states, etc. So we think it's really important that we enable this type of analysis going forward. Anyway, sorry, that was a bit of a tangent, but this is an example of where you can get proteogenomic insights from Xenium. So this is a clear cell renal cell carcinoma sample. This is RNA-based clustering with a 477 gene panel. And you can see there's a couple of different clusters here, just annotated by the color of the clusters, two different types of tumor cells that have been identified by RNA-based clustering. Again, this is the H&E image here. You can see that they've all been marked independently by a pathologist as being grade 2 invasive tumors. But if we do protein-based clustering with our protein panel, we actually see there's a bit of a divergency. This cluster at the top that's outlined in purple looks different by protein-based clustering than it's been separated out as a different cluster by protein than it had been by RNA with this particular panel. And if we look now and dive into the protein markers we've got, we can actually see that this is extremely low in CD138, which is an important tumor marker. Knowing that now, we can go back and look at individual genes and see what's up, what's down, what's driving these differences. And we can see that there's a couple of different markers here. These are soluble factors, DPP4 and SOS-DTC1, that are usually involved in things like stem cell renewal. So the sort of markers that you probably don't want expressing in your tumor. But I think this is just a nice example where protein and RNA were able to sort of further subdivide and further identify new phenotypes within that sample. And Xenium protein panels have been validated across and pre-optimized across a wide range of tissues. These are just some examples of human tissue that we have this beautiful data on. And with that, I'm going to stop talking about Xenium. I gave a brief overview of the Xenium system. I think you'll probably hear a bit more about this coming later. But I wanted to emphasize that coming very soon is this single RNA and protein workflow integrated that will be coming very soon. So I just want to sort of end with this slide to sort of say these are the things that we are motivated by at 10x. We really want to enable low cost and large scale projects. Performance is always top of the list for us. We want to keep expanding single cell and spatial capabilities for you guys, for the work that you're doing. Together with this, we're going to always commit to building our software in line with our assays as well, so that analysis is never a bottleneck. But ultimately, we really want to just build products that will increase your insights, enable you to answer questions that you couldn't answer before. All right, with that, I think I'm right on time, and thank you very much for your time. Are we doing questions or are we just plowing through? Okay. Yeah.
Custom add-on protein to the panel?
Not at this very second, but that's something that we're working on. It's on the roadmap. We should speak offline, but that's the direction we want to work towards.
Proteins that are not part of the panel, and
yeah.
like, okay.
For anyone who's ever developed a panel for anything, it's never enough. So no matter what you've got, there's always need for more. So we're definitely adding custom proteins to this is definitely something we want to work with people on. I will say custom protein in this type of context is challenging. So I think we need to be prepared that we're not going to pretend that this is going to be easy and you can just drop in whatever you like, because I think anyone who's worked on these types of tissues knows that is not necessarily the case, but we want to be upfront about that and work with people to enable this.
Thank you. And in the 3', are there any proteins on that? Or?
Not immediately at launch, but I think people are already Visium 3' V1, people are running protein on that right now. There's active work on doing this for HD, and I mean, there are already some people that are playing with this with total CK antibodies, and yeah. So I think watch this space there. Yep, please.
Yeah, I might have very stupid questions. So, I mean, in spatial biology, are we interested in the resolutions? Now, if I'm interested in the dynamic of, let's say, mitochondria, like fusion, fission, whatever it is are we going to get to that point where 10x?
Good question.
Dynamic of that, or like a Golgi complex or something like that.
I mean, right now we're focused on tissues that are very much fixed and very much dead. So what monitoring live processes is not something that we're working on, not something we've got products on right now. I agree. I think that would be a very interesting direction to move. Do you mean in live cells, actually watching?
Just live cells, like even in FFPE, can the resolution get to like that?
Well, I think, I mean, FFPE is going to be capturing a very defined, precise state. And I think I didn't mention this, but our Xenium assay is completely non-destructive. So people can and do take our slides after running Xenium and do cyclic immunofluorescence, multiplex immunofluorescence, immunohistochemistry. Like these things all happen afterwards. So your tissue is not destroyed. So if you have very precise stains that measure these things, you can go ahead and do them and take that slide and put it on a high resolution microscope and then map those images back onto your Xenium data. So that capability already exists.
Subcellular, Xenium is subcellular.
Yes.
But can you look at proteins at the Golgi level?
Yeah.
That's what can we look at the sub, the organelle level? That's what you were trying to ask?
I mean, I think the protein stains look indistinguishable from immunofluorescence. So, the resolution that you get from immunofluorescence is the resolution you would get from our in-house protein stain. You can also do post-staining and do actual immunofluorescence and look on your own microscopes too.
Not to sort of jump in uninvited.
Sorry.
But your question really comes down to the technology that you're going to employ to address the size of the Golgi. So you're limited by the resolution of the system. So as he pointed out, certain microscopes have the capability of going to 60x, 80x, where you can see a Golgi. Other technologies are limited in that. It really just comes down to, but the limitation of the microscope is how many markers can you look at? So it really sort of depends on how many markers are you looking at for that level of resolution to be able to see something the size of a Golgi or an exosome, etc. So theoretically possible. Just you have to match it to the technology and understand the limitations of that technology.
And there's also throughput trade-offs, right? The higher resolution you're looking at, the longer it takes you to image what you're doing. So, we are trying to emphasize the ability to scan very large areas. And, we could have used a spinning disk confocal, but if you do that, you're not going to be able to process thousands of transcripts in you might have one experiment that runs 5 weeks. And I don't think anyone wants that or more. So we're trying to keep the workflow to the point that you can use this and generate a large amount of data over time. But you can always take these slides and go and do super resolution microscopy afterwards or whatever you need to do, and then map that information back to the transcript information.
Does a Hyperion do that?
You can't detect what you have.
No. Okay. So there's somebody called Sincel that is, I think.
Our resolution, our technology is about 0.5 microns, so we're subcellular.
Okay.
So, but looking at mitochondrial Golgi, we're not quite there yet. But we are, we're not in this talk, so I don't want to.
It's all right.
Technology have to address it after the talk, but I don't want to take away from the light of people who are.
Nah, guerrilla marketing is great. Look, it's awesome.
But we have data, as you pointed out, using the anymore platform.
One more last question. There are other competitors of in the space who are doing whole transcriptomics at the subcellular level, in situ base. Is that the direction that 10x is going, or would Visium HD would be a good alternative?
I mean, I think to give this sort of very political answer, we are all invested in our Xenium and Visium platforms, and we're going to be continuing to drive these forward in every direction you can possibly imagine: speed, throughput, plexi. So you can read between the lines there, but yes, we'll be expanding our offerings as time goes by. I would point back to that slide that I had that had 8 different bioarchive papers that show performance.
Yeah.
If the performance delta is strong at one level, adding additional transcripts to that is unlikely to make the performance any better.
We're just worried about how long is the machine going to run.
Yeah,
For whole
right.
transcript.
Well, I mean, we're really mindful of this, and I think I've made this clear a couple of times that, as we do look to increase the panel density, we don't want a linear increase in time that it's going to take to run that, right? So if it takes 4 times as long to run 20,000 as it does to run 5,000, we're at 6 days for 5,000. Do you have any appetite for running your instrument for 20 days? I mean, probably not. Or the amortization of that instrument, it doesn't make business sense for you to do that. So no, we're very mindful of the fact that that's not, it's probably not a mass market product if it's going to take forever to run a single sample.
Thanks.
You're welcome. Sorry. Okay. Thank you, everyone.
Thank you, Peter, for the informative presentation. Your insights into expanded capabilities of Visium HT and integrated Xenium RNA and protein detection workflow offered a glimpse into exciting future of spatial biology. Appreciate your expertise and clarity with which you shared these advancements and roadmap. Next, I'm moving to the next speaker. Now, I'm truly delighted and proud to welcome our very own Dr. Vidyodaya Sundaram, VP of Business Development at BioChain Institute. She has been responsible for spearheading BioChain's journey into spatial over the last several years. In her presentation, Precision Validation in Xenium, BioChain Spatial Solutions, she will introduce us to BioChain's suite of spatial capabilities and walk us through BioChain's precision validation testing on 10x Genomics Xenium platform. Without further ado, let's talk a little bit about BioChain Spatial Solutions. The stage is all yours, Vidya.
Thank you, Nidhi. Thank you so much. So we got into spatial biology a few years ago when we got in, and we still feel it's a complex platform. The data is very complex, but our job is to make it simple. So this is our slogan, and also Final Frontier. I'm a huge Star Trek fan. So that's why I brought in the, Final Frontier. And the other side of the t-shirt is Discover the Unknown. So just like what Picard would say, we are very excited to be part of Cell Bioscientific. We were recently acquired by Cell Bioscientific, and our new CEO, Charlie, is here. And just to give you a brief agenda, so we'll just go through who BioChain is, and we'll talk about our capabilities in biospecimens, and then our spatial biology services. We have been here for more than 30 years. Last year was our 30th anniversary, and we have been having multiple customers coming to us doing various different assays and also biospecimen projects with us. We are an ISO company. We are ISO 9001, 13485. That is a very critical part for quality control for all our services and products. We are a global company in terms of we have distributors across globally, and also we have more than 1,500 products and services. These are our core capabilities. We started off as a biospecimen procurement company, so we do have both archival and also procurement services. We also have real-world reference and control samples, which means that we go about extracting RNA, DNA, protein from the samples that we have internally, from mostly oncology or neurodegenerative tissues. We also have automated solutions. And we have our own extraction and purification kits, and there are a lot of OEM partners who we work with providing our samples. And the most recent technology or service we started was spatial biology. So I just want to give you a little bit of what we are doing in terms of samples. So if anybody over here looking for samples, and we've been working with all the partners over here from Leica to Standard Bio to 10X and others in terms of providing good quality samples. So we do both prospective as well as retrospective samples. We provide FFPE, OCT, fixed. And then I think what we really do well are these matched oncology samples with for MRD and for early cancer detection. So that is something that we, we have multiple collection sites across the globe, so we can go out there and get samples from various ethnicities and help either assay development or part of preclinical assay development. And for example, just to show you some of like what type of samples we have both human and also a non-human primate, mouse, rat, and plants. We have normal tissues, disease tissues. We worked with Leica for Alzheimer's, and also with 10X, we've had many of our tumor tissues have been used while validating their assays. In terms of donor information, we have both longitudinal. We can follow donors for more than a year and collect samples at every at various time points. We have pathology diagnosis, age, weight, BMI, smoking status, LRAD scores, a lot of these additional information that are required. We can provide that, and we can provide from the same donor plasma, Buffy coat, FFPE, primary tumor, adjacent normal, frozen frozen adjacent normal, and frozen tumor. So we have quite a good list of samples. So now coming to spatial biology. So I've been at BioChain heavily involved in bringing this technology on board. It's been a huge learning for me personally and for the entire team. So in 2020, when the method of the year came out to in Nature as spatial biology, in 2021, my CEO Grace over here, she came and asked me, "Is there anything interesting out there so that BioChain get into?" And it took us a while, and then we decided to get into spatial biology. The reason is we had the tissues first. We understand tissues, and also we have a great histopathology team at BioChain. And also it requires, as Charlie mentioned, the genomics part too. So even though we don't do the sequencing, that can be easily outsourced, we also do a lot of genomics capability in-house, building library preps, doing DV200 and all those things. So this was all within the same roof. So most core facilities, you might have to go to different buildings, and this was all under our own BioChain. And so that kind of helped us to say, this might be a technology where we can get in and easily, onboard, as I thought, but it wasn't, because as Peter mentioned, the first version of Visium was not easy, where you had to place those samples on those slides. It was quite challenging, but we were glad when we got the Site Assist. It made our scientists' life a lot easier. And then in 2023, we were the first to get the Xenium. We got into the whole, ideology that, doing subcellular location is very critical, because spatial is basically looking at you want to do location. So you're really, that part is very critical. And last year was a very, very exciting year, because 10X, it's hard to keep up with 10X. They come up with new products every 3 months. My scientists are like, we have to do SOPs for each one of these technologies we onboard. It's not that we just do it without, having our internal SOPs. That's very, very important for us to maintain our quality. So last year we were able to onboard Visium HD. Xenium multimodal, and Xenium 5000 genes. So that was quite a lot of work by our team. And this year we want to onboard the HD3' as well as the proteomics. That's something that a lot of our customers are asking for, and we are excited, and we will be one of the first, CROs, to get, once the proteomics is out, we will also be involved in it. So as I mentioned, why BioChain? Why come to us? First, we are, we got into spatial biology at an early time. Our learning throughout the last 3 to 4 years has been tremendous. Our SOWs and SOPs have grown as we've gained more experience. We have project managers for pre-sales and post-sales, and I'll talk about that. We are fast. We can turn around things within 3 to 4 weeks, which is, when you go to a core, that might take a long time. And we work with, since we are ISO, we work with clinical samples. So people can send clinical samples. We will trace the samples. We will make sure that these samples are handled, carefully and, properly. And we work with all kinds of samples, biopsies, organoids, and we can also, work with biotech and biopharma too. And the last part is data analysis. That has been the biggest bottleneck with spatial. The tremendous amount of, data that comes out of these things. We have built many partnerships for data analysis, as we go. So when a customer comes to us, right? So what is it that they are asking for? So the first, spatial, why come for spatial? They are interested in location, just like real estate, location is the most critical part, right? So the first thing when they come to us, we have an initial discussion. In 2021, there was a lot of learning that we had to learn, and 10x helped, and also the customer had to do a lot of learning. But as we started to progress, we've learned that customers now know, understand spatial biology, understand some of the initial, questions when they come to us. So that's a huge difference that we've seen in the last 3 to 4 years. So the first thing we ask is why? Why do you want to come into spatial? There are multiple other technologies, much cheaper, like bulk and even single cell. And then, then we decide, do you want to do targeted or discovery based? Then the next question is, what type of analytes are you looking for? Is it RNA or protein? In case of RNA, are you looking at whole transcriptomics, or are you looking at targeted gene panels? Do you want to do some custom gene panels? Are you going to look at RNA and proteins, or do you want to just do proteins? And now with the acquisition with Cell Bio, and then we have our partner, Aspix, so we can do a lot of these things, and we are in a much stronger position. And the last thing is, where are you looking at? What is it that, are these analytes? Where are you looking at the subcellular level, or the ROI level, or clusters of cell, or is it single cell? So once we understand what people are looking, then we start understanding the journey of customers, in the sense that if you come to BioChain, you can start off from discovery all the way to the targeted small plex. This is something that I think we stand out, because not too many CROs out there can start off with single cell, subcellular, which is over here, 10x Xenium, and then if people just want to do discovery, just do Visium V1, or if you want to do single cell, the whole transcriptomics, then do HD, and then move down to multiplex IF, 3 markers or 5 markers, or do IHC. And now, as I mentioned with Aspix, we have excellent advantage in doing this. So again, then we discuss about what type of tissues does the customer is interested. Do you want to bring in tissues from a clinical trial, or are you looking at some preclinical samples, or do you want to use some of BioChain's repository samples? And then we can do all the tissue processing. So, people do not want to touch some of these tissues, but we are happy to work with these tissues, especially OCT tissues. And then they can select the assay that they are interested in, whether it's Visium, HD or Visium, V2 or Xenium. In Xenium 2, you can do custom or 5000K. We've actually done more than 10 5000K projects, which is around 5 to 6 days each project of running on the Xenium. So it comes to around 2 months of running the Xenium at BioChain. And we are continuously doing these Xenium projects. I think we've done more Xenium projects than 10x themselves have done, maybe with customers. But we have been churning, keeping that machine running. And now we have 2 machines in-house just so that we can, help our customers push through these projects at a much faster pace. We also do TMA construction. This is a TMA construction and OCT construction that was done at BioChain and validated, used for assay development and validation at 10x. And I think this is very critical. As I hear from pharma, as more conversations we have with pharma, it is the quality of the data that is very critical, right? So we really understand how critical quality of data is. So when we do these ISO, part of the ISO is building these SOPs. We just don't take the 10x's protocol and then just follow it. We have improved it. We've added checkpoints. We've added third-party checks, even to do a little click on the PCR to make sure that it goes well. Because initially, the first couple of times, we didn't do it well. The slides broke. We do not want to, thank God it was our own slide. So we have built these workflows so that, when we run these samples, we have good quality at the end so that we can provide great quality. And we have an internal pathologist who can look at the morphology, identify if people are interested in ROIs. We can do RNA and analysis, quality analysis using for DV200 or RIN scores. And then and the slides, right? So especially Xenium slides, they have these fiducial markings. So we make sure that those tissues are within, they are not touching those fiducial markings when we build and place it, or when we get samples from outside. So these are at every point, our team will check, will take photographs and communicate with the customers. And the other thing that has happened, and there's a potential, is cracking sometimes the quality, especially clinical samples. People don't have biopsies. They are so small that they do not have control. But we make sure we've made some modifications in the baking steps so that the peel off or cracking and other issues that would damage the tissue is minimized. And then we do a data QC, and then the data is presented in a transferred in a very secure manner. So we take quality very, very seriously. And as I said, we want to maximize these assays are not cheap. We understand that these assays are, take a long time, and both the reagents and the hands-on work is sometimes can be very, very tedious and requires a lot of effort. So what we try to do is we maximize, so we can build TMAs. We tell people, if you have many of these samples, put it on so that we can just, run more samples and minimize the cost. And we even have a little cheat sheet over here to say, depending on how big your core is, if it is 1 millimeter, you can add 27 cores, and so on and so forth. So we really work with our customers to help them decide from which platform they are interested, all the way then understanding which panel or WTA, the whole transcriptomics, and also the sample preparation. So that is the strength of BioChain, is to have that whole end-to-end solution in terms of finding, addressing all the different parts. This is our spatial team, service team. So as I mentioned, we have 2 project managers, pre-sales and post-sales. And then we have very clear roles for each team, the service team versus the pre-sales and the post-sale, and they are also certified. So as we started doing these projects and we had more conversations with the pharma, people kept on asking us, how good is the technology? How good is the platform? You know, and we kept showing 10x's data, but people were not convinced. They wanted us to show it at BioChain. So the trick was, can BioChain do this? You know, your scientists at BioChain, can they replicate some of the same things that you know, people see from 10x? So that's when we said, okay, if we really want to go and impress pharma customers, we need to prove them that, you know, the reproducibility, the quality of data, technical, you know, we've done technical replicates. All these things are very critical, so that we have to go show our customers that we can do these assays in-house. So we started study design was we worked on Visium V1, reproducibility, sensitivity, and also we wanted to look at concordance with another assay, Visium V1. So we have, you know, we have a ton of samples in-house. So we used our own breast cancer, colon cancer, and lung cancer tissues. And we used the, you know, multi-tissue tumor panel for Xenium, which is 377 gene. We made a TMA. Then we did serial sections. 2 different slide operators, because the throughput for Xenium is 2, and we ran it on 1 Xenium on 1 day and looked at the reproducibility. And look at this. This looks perfect, right? The R is around 1. So this is this gave us a lot of confidence. This was the first piece of data that we got that gave us a lot of confidence. So this was serial section, reproducibility, same run, different operators, great results. Okay, our scientists have great hands. We now, you know, I can confidently go out there and talk to customers that, hey, you know, we can do this. The next is we wanted to do a little bit more deep dive into some of this, some of the data. So we looked at median transcripts, and, you know, if you see, obviously colon is a little bit low because this is not a colon specific a panel. This is a multi-tumor panel, but it looked pretty good. Then we also did H&E overlay. We had a pathologist who was able to identify immune cells, blues immune cells, yellows, purple is carcinoma, and then we have other types of cells that people looked at. We wanted to do the a pathologist was able to annotate it, and then we looked at stroma cells over here, immune cells and tumor cells, and we were able to identify these cells. So that was the next step. After this, we wanted to look at genes that are specific to those regions, those types of cells. Again, we were able to show that there are specific genes that would mark immune cells and also stroma and tumor cells. So there were specific genes that will be expressed only at these types of cells. And we focused on the breast cancer tissue. We had 3. We looked at breast, lung, and colon, but we wanted to focus on the breast. The breast cancer tissue that we used was a ductal carcinoma in situ, stage 3, stage 2. And if you look at over here, majority of the cells are DCIS cells. So that clearly showed that the Xenium was able to pick up cells that are specific to this cancer type, which is the DCIS cell types. And then there were some immune cells also that they were able to pick up, which is CD4 and CD8. Then we wanted to look at the spatial context of it. We're doing spatial biology here. So all the stuff, maybe you could do it on single cell and things like that. But here, what is critical is spatial. So can we now go and look at specific parts, within this breast tumor, which is spatial, tumor as well as immune cells. So, and this is something we identified 20 specific genes, and we were using the volcano plot. We were able to identify that ABCC11
is highly expressed in tumor, and CD8 is highly expressed in immune cells. So these are just two regions that we identified in this specific tumor. This is a tumor region and this is the immune region, and you can see the tumor has mostly tumor related genes that are being highly expressed. You have the AGR3. I'm not the specialist over here, and then EpCAM. While on the immune side, you see mostly the CD2 and this CCL5 and so on. So this clearly showed that you can spatially identify specific regions through the xenium data, and you can also find genes that are highly specific to this. The next part is the cell distribution within those two regions, right? So this is the tumor region, this is the immune region. What type of cells are being distributed? Again, we were able to see in the tumor, mostly DCIS, and then that again correlated, and in the immune region, CD8 and T cells. So this gave us a lot of confidence that there is specificity in terms of genes, and it can also find cell type that are very unique to these regions. So these were the key findings: is high reproducibility in our hands, morphology consistency from serial section. So the data I showed were all from serial section, so there was not much difference between them. So, great morphology consistency. Gene expression was also pretty consistent between those two serials, even distinct cellular between tumor and also immune regions, and operator independence, right? So it didn't matter. Sometimes, you could use, we also did some, operator independence, like different operators, and we were able to see that too. So the next part is people came to us and said, hey, can you do IHC? Can you do immunofluorescence? And 10x says, these xenium slides are non-destructive. We wanted to check it out. Is it really non-destructive? And in our hands. And we do H&E so that you can overlay it, and people can, can really understand between the overlay and the xenium, and also IF. And we did an orthogonal technology using understanding how Visium and xenium work. In terms of IF, we did a proof of concept with Spatomics, and Shashank is going to talk more about the technology. And we gave a couple of our xenium slides, and they were able to look at, I think, post-xenium 10-plex protein, right? So he's going to talk about that. So I'm going to skip that part. So what we did was we took the xenium slide, and the breast tissue, we did Visium on it. This is not Visium HD. We did was Visium V1. So this is comparison between xenium on this bottom part, and then xenium over here, and then Visium. And you could see the clusters very similar in terms of, even though this is 55 microns, we could see some, the genes, EpCAM, and then you could see CD8, CD4, and also specific tumor hotspots. So this gave us a lot of confidence that, some of the results that we are seeing in xenium can be correlated in another technology. Then we wanted to look at the sensitivity between xenium and Visium, not HD again. So we knew that Visium is not going to be as sensitive as xenium. And again, we saw that the xenium detects obviously more, even though it was only a 377 gene panel. It was approximately around 10 times more sensitive than Visium V2, and it was able to pick up very rare genes, low expression genes, and also rare cell types too. So we were confident that xenium, if people are really interested in looking at some rare cell types or low expression genes, that's something that xenium would be a great platform. The next part is people wanted more. People wanted from multiple days, because we are getting clinical samples that are taking sometimes more than 2 to 3 months from the time they start getting the first sample set to the time they, we get the second sample set. So people were interested, how reproducible, is Xenium in your hands. So we worked with the 10x. And they gave us the reagents. We again used our repository samples, and we made another TMA over here, 4 different carcinomas: stomach, colon, breast, and lung. And we compared Xenium to Xenium reproducibility from the first day to another day, and then Xenium and Visium concordance, and different days, same day, different operators. So we really went ahead and did this. We are still digesting the results. It's just so much results. We need a bioinformatics, an army of bioinformatics people to really work on this. So I'll just give you the basic study design. So here once we made the TMA, we made sections. We call it the first batch. We did Xenium, and then the second section after this, the serial section, was the colon. And then we made another couple of sections. So these were done a little bit further apart, but done completely different days, right? These two, so batch 3 and 4. And then we did another batch for 4, which was actually, we used some multimodal segmentation. By then the multimodal had come out. And so we are now, we've done all of these different runs, different operators, very far apart. There's a good distance between this batch 1 and batch 4. It's more than 20 sections. So I'm just going to show you very briefly what we went through. So this is batch, I think this is batch 1 and batch 4, and you see the multimodal works really well. It's, it's more specific. It doesn't, because of the multimodal segmentation versus nuclear segmentation. So that was, that made us a lot more confident. And now we suggest everybody to go use multimodal segmentation. And this is a UMAP of 3 different batches, and it looks perfectly aligned. And also we also showed how consistent our, from batch to batch. And then we also saw some gene specificity here. This is a gene, tumor suppressor gene that is, that you can see across. I think this is the stomach, colon, breast, and then lung. And you can see a breast-specific gene, lung and gastric specific gene, and CRC and lung gene. So again, Xenium can do specific genes across each type of indication. So again, key findings, we are still working on the data. We are publishing each year, when we go to SITC or AACR, we go and present these posters. So robustness, same run, different, different days, different runs, sensitivity across Xenium, and also gene expression and cell distribution. So it works really, really well. We also offer geomics nanostring DSP, just very brief. Again, it depends on what the question people are asking. Are they looking at whole transcriptomics, ROI based, and also they also have a large immuno-onc protein proteomics panel. So we have not done the proteomics panel, but it has been done by nanostring. So we're very confident that we can do it. So we used our TMA, breast cancer TMA. We looked at 2 different markers, and we were able to see how sensitive geomics was. So this is just a brief outline of the various technologies we give: direct placement, site assist, HD3 is something we will be providing soon, Xenium and DSP. And each one has its own flavor and advantages. So it really is, it depends on what the first question, like what I mentioned, is people are interested in discovery, the what, where, and where, that is what is critical. So when we talk to a customer, we provide our entire set of assays, platforms that we can provide at BioChain, and it gives a lot of confidence for the customers to decide which platform would be the right one for them. And then as we move forward, this is a key strength in terms of histopathology and genomics under one roof. ISO process because quality is very critical. We have project managers, we have very, people who can handle clinical samples, data security. And the future roadmap is in-depth analysis. We are building, we're trying to build more partnership for computation and proteomics. And now we can also provide multiplex IF and IHC, and Visium HD 3' is something that we are looking forward to onboard. This is our special spatial team. We have Elam, and then Tommy. Elam, you want to raise your hand? Tommy, and then Jing Hao, who is our intern. He's been with us for the last 3 years. He's done a lot of analysis for us. And then we also have our fabulous histopathology team and Rikita, who's unfortunately couldn't make it here. It's a solid team. So we've done a great job, and I'm very proud of our team. Thank you so much. Any questions, or do we want to take a quick break and then come back? Or Rick?
That's a lot of questions, right?
Yeah, Rick, you want to start?
Be happy to. Whatever the crowd wants.
Thank you, Vidya, for an insightful presentation into our spatial biology services, QC approach, customer journey, and Xenium precision validation testing. We appreciate your leadership and clarity you have brought to BioChain's spatial solutions. Over to next, let's dive into the world of multiplex data visualization. I'm happy to introduce Rick Hale Chapdelaine, spatial biology specialist from Leica Microsystems, who in his presentation, Omics with Spatial Context, is going to share some cutting-edge methods for generating multiplex 2D and 3D spatial data, querying within those images for deep multi-omic insights. With over 7 years championing self-dive multiplexing and a rich background in academic research on the cell cycle, Rick brings unparalleled insights into the tumor and tissue microenvironment. Rick, the stage is all yours. Over to you.
Thank you very much. And thank you all for coming. And thank you so much, BioChain, for inviting me to come and present. I'm a little disappointed. I had shared that I had met my wife doing Bavarian folk dancing in our Get to Know the Researcher, and somehow that didn't make it in there. So, oh well. Had to make sure you learned this. Let me go ahead and grab the little pointer here. Okay. This gravity is feeding against me on this slide. Multi-omics in spatial biology. We'll start there. As you can see, we have a quick development of these different technologies becoming our methods of the year, where in 2020, one of the other methods of the year was our multi-omic single cell analysis. And last year, 2024, method of the year was spatial proteomics. And we'll kind of walk into kind of the evolution of that and kind of where we hope to walk off, not into the sunset, but hopefully into new dawns. So the kind of first push for trying to understand the need for spatial biology was this development of our lack of understanding of cancer. We had wonderful immune therapies. They were cures, but frequently they didn't work. I actually have 3 brothers that are survivors of cancer, 2 of them because of different immune therapies. So I'm very fortunate in my family that we're part of this 30%, as this little chart says, that are responding, but we want to know more about the ones that are not responding. So let's take a look at the tumor. We want to understand more about it. And I think there's 3 different things that Vidya kind of commented on. We could start with bulk, but then we have kind of low signal to noise, right? We can learn a lot of things. That's kind of how some of the first mutations were identified. And then we can start moving into more single cell analysis, where we get high signal to noise, but no longer have any spatial context. So then we want to move into spatial. Hopefully this will kind of carry this across. So we want to get better signal to noise. We go ahead and we have a single cell. We can do protein, which would be your flow cytometry. We can move into single cell seq with your RNA, DNA, and we can get all of these pieces of information for our multi-omics on a single cell. But that doesn't tell us what's happening within the tissue and how they're interacting with each other. So we actually want to move into 2D tissue, and this is protein using antibodies looking at a variety. I'm drawing a blank on which cancer this one is at this point in time. But now we can go ahead and look at all of these different types of well, we like to look at all of these within the tissue, and we can currently look at just a few of them at one shot, but I'll talk about how we can look at more. But then what about 3D tissue? So this particular sample came out of Ron Germain's lab, and it isn't a huge number of markers, but I think we can tell that when we're looking at a 2D section, we're really missing something. This is a lymph node, and you can see the vasculature in yellow. You can see all these immune cells and a variety of other cells in here. And there are certain, they're not a niche per se, but vascular regions that have different cells in them than we would have anticipated only looking at 2D sections. It was kind of missed in earlier studies. So it'd be really helpful to move into 3 dimensions and be able to see what is the relation. Also, the lack of missing rare cells. There's a lot of benefit to looking at things in 3D. I mean, in fact, last time I checked, I was 3-dimensional, as are all of you. Now, this answer is, oh, he left. The guy that was asking about organelles. So in 3D, I was talking about how do we look at the cells within each other within a whole organ or tissue section, but then how do we even go subcellular and look at relationships of proteins or organelles within cells in 3 dimensions? Luckily, Leica has a lot of solutions to cover these. What's really I'm proud of being at Leica because we're the only real microscope company within this space. So we understand imaging, we understand optics, and we know how to make really, really good data. And we'll go into that. So kind of the different technologies I have on here, and I'll cover them a mile wide and only an inch deep. If you want to go really deep, come talk to me afterwards. We'll talk about cell dive. We have customers that are going over 150 markers. We kind of say 60 plus. It really depends on the study and how they're working. But this is for our 2-dimensional high-plexed imaging, probably off 325 nanometer resolution on each of the pixels. We have Spectraplex, which is a confocal version, which has complete control of resolution as you would with a confocal, and the ability to do thin or thick specimens, and we can do 15 at one shot. Very helpful for looking at a lot of 3-dimensional structures or getting higher resolution for your organelles. Now we have this beautiful data that we're known for being able to collect, but then what do you do with it? Well, raise your hand back there, Wanyung. Okay, she's the queen. I'm really bad at titles, so I'll call her the queen or guru of our analysis platform, AI Visualization Image Analysis Platform, for actually being able to take that data and create numbers and insights from it through a number of graphical methods. And not just spitting out a graph, but being able to take that graph, the table, and the data and interact with them all linked to really generate insights. So it's not just how do we spit something out, but how do we query the data for more information. Now, let's say we don't have sufficient markers for evaluating what's in situ on that slide. We can actually take what we can identify with AVIA, those little cells or tissue regions, export those shapes as an XML file and bring them into our laser microdissection, cut those out as purified tissue pieces or individual cells, and really bring them into a number of downstream analyte analysis. So I'm going to really kind of highlight a very broad approach to looking at a variety of ways to delve into the biology in maintaining spatial context. Now, does one of these click forward? No. I should have practiced this mouse beforehand. Okay. So cell dive is the first one I'm going to talk to. That was on the upper left. It was developed at the GE Research Campus, actually, there's just a podcast from Fiona Ginty recently. The first concepts that they were talking about was around 2005, 2008. They started developing it. The first paper was the Gertis paper in 2013. No, that's the first paper there, but the 2013 paper is probably the one more known. And then it was brought along. I kind of jumped on board about 2017. And then We were acquired by Leica Microsystems in 2021. So I've been with this product for a bit. There's a lot of iterations through this whole process where we've really looked at how do you go from digitization, how do you go from multiplexing cancer to multiplexing immune cells, to how do we think about reproducibility and robustness within, this isn't a clinical instrument, but within the clinical, at least developing that for the clinical space. And there's been many iterations in working on how to actually accomplish that. And hopefully I'll review a bit of that here going forward. So here's the basic process. So on the left, this is where I'd call BioChain, because I'm going to need a single tissue section on my slide, or maybe a block to create serial sections. And what we're going to do is it's standard 1x3 slides. We don't have expensive consumables. So what we really want to do is we want to create robustness, reproducibility, keep flexibility high, and cost down, because spatial biology is not a cost it's not a low cost endeavor, right? We select our regions, and then we go through an iterative multiplexing immunofluorescent process. So this is optical fluorescence. And there's a number of ways to do this. It's a confusing market space. And what we do is we use directly conjugated fluorophores onto the antibodies. This is probably developed way before we even had secondary antibodies. So it's a tried and true methodology. It's cheap, it's easy, it's gentle, and it's not complicated. Oligonucleotides can fail. This really makes making your panels quite robust and simple. We image it. We don't strip off the antibodies, because if you're stripping antibodies, you're going to be stripping off epitopes. And so what we really want to do is leave that antibody in place and gently inactivate the fluorophores. Image the autofluorescence again, right? So we've talked a lot about FFPE, archival samples. One of the challenges for fluorescence imaging is there's a lot of autofluorescence there. Standard BioTools deals with it by using mass spec. There's no fluorescence. You don't have to worry about it. We deal with it optically. We collect the image every single round because it decays nonlinearly over time, and we want to have reproducible data of your signal of biomarker, not artifact. And so we collect it every time and subtract it, and so we get very high signal to noise. We go through a two-part registration of imaging the nuclei before we image the other markers for looking for rotational effects, and then we start imaging it, and then it goes into the background. We have an imaging process that happens in the background of lateral alignments and autofluorescence removal, and then analysis. In our discussions with a number of people doing this work, the bulk of your time is going to be in this analysis. This seems onerous, this process that we're helping to deal with, but we want to make sure that when you get to analysis, what you're analyzing is actually has high confidence levels. So we really work hard on making sure that the data is reproducible and quantifiable, so that when you spend all this time on analysis, you're getting high signal to noise data that will give you really unique insights. So why multiplexing in here? Well, we can go ahead and layer over many different questions on the exact same tissue section. So we can layer over the sample the ability to look at cancer immunity and different progressions of the cancer. We can look at what's happening with the metabolic state within a tissue. This is an adenocarcinoma, colon cancer. And then we can actually look at things like what's happening with the cell death. I'm going to move on to another type of cancer, pancreatic ductal adenocarcinoma, which is a very aggressive cancer and is recalcitrant to a lot of treatments. And with these sets of markers, we can kind of move on and ask some questions about, or at least make a hypothesis that perhaps these hypoxic regions are selecting for cells that are resistant to cell death and therefore resistant to treatment. So we can start looking at some of the markers and the relationship of these markers in space to start answering this question. So across this whole bottom here, you can see all the markers here in the stendogram, but even just with a few markers down below, we can kind of map the tissue to see what is normal pancreas. The rest of this is all tumor, and then it's kind of subdivided into hypoxic and normoxic tumor. So let's go ahead and take a quick look here at what's happening with GLUT1 as a reporter for our hypoxia. And we'll see up here, cluster 4 and 6, we see that these are normoxic cells, and they separate quite a distance from those. If we clicked on those, we would see they map to the tissue that is normal pancreas. So this is just healthy cells. We map this cluster number 1 from this UMAP. We can see that if we came over to the tissue section, it would map to our normoxic part of the tumor here. And then if we were to go ahead and look at this hypoxic region, cluster number 3, so we see the expression pattern and the location of these cells is here. So that's the hypoxic region. Now let's go ahead and ask a question about what's happening with cell death. So we'll take a look at this back one, and we can see here these cells, which physically map to the normal pancreas over here, we have normal cellular turnover. It's normoxic, some cells are dying, some cells are living and doing their normal day-to-day activities. We look at cluster 1, and that maps to this space here, but we see that apoptosis is quite high. So there's a lot of cell death in this region of the tumor. And then if we look at this particular cluster here, that maps to the hypoxic part of the tumor, and there's low cell death. And so that kind of matches this hypothesis that perhaps within this region of the cell, we're selecting for cells that evade cell death, and that might be one of the mechanisms for cells being recalcitrant to treatment. Now, that's not very good for this patient, but then another thing to check is where are the location of their immune cells? So if we look at the clusters of these immune cells, here they are, cluster 8 and 9. If we map them back to the tissue, it turns out they are all, in fact, residing within the normal tissue. So sadly for this patient, they're not a very good, they're a rather of an immune desert within the tumor, and so they're not a very good candidate for immune therapy. So those are the types of questions we can ask with these types of markers on this tissue. So CellDive, here's all the different parts of it. We have validated antibodies. We're at over 400. We do not sell antibodies, but we provide the clones and the vendors. So what does this do? This keeps your flexibility high and your costs low. The whole CellDive process is this chemical inactivation, and we do have a 2-part antigen retrieval. We want to make sure that they're completely accessible to the antibodies, the different antigens. And then we have software and hardware that work together to create beautiful tracking of your different projects, whether they're synchronous or asynchronous projects. And then we have the beauty of being able to do analysis of that data for clustering your cells into phenotypes of interest based on the very accurate segmentation of those cells. And then we can use a number of spatial relational tools in various charts. So I mentioned antibodies, and the reason I want to do that is talk about this flexibility. We have about 400 validated antibodies. We can help you validate your own. You can use third-party kits. I mentioned we're working on FFPE predominantly because that's the hardest as far as antigen retrieval and getting rid of autofluorescence. However, you could look at other types of tissue. Also, this instrument is agnostic to what you're going to attach these fluorophores to. So we have groups that are doing RNA scope and molecular instruments type of staining for what is happening with the transcriptome in addition to the protein. We have groups looking at post-translational modification using lectins for binding their cells with these fluorophores, and you can cycle these different colors on, off, on, off. It doesn't matter what they're attached to. So reproducibility. We have a calibration plate that we put on the instrument that goes to a ground truth state of intensity calibration so that you'll get the same signal today, next week, next year, and across multiple instruments even. And that's just one of the calibrations. There's a whole series of other calibrations that allow us to get this seamless image from edge to edge across a large field of view. Within our robotic automation system, we're looking at 17 by 37 millimeters of space, or 6.3 square centimeters of tissue. Now, when we're doing spatial biology and we're phenotyping, we are measuring by intensities of biomarkers. We don't want seams across your tissue that are going to show variations of intensity that are reflective of artifact, not reflecting of the tissue, actual biology. And so we really work hard on creating artifact-free imaging across your whole side. I didn't bring the poster with me, but we do have a really beautiful reproducibility poster. So that's across the whole slide. We're doing iterative imaging over. A lot of people have concerns with how well do those align. So here we have in blue Ki67, which is from round 1 of biomarker staining, and we have DAPI from round 3 of autofluorescence imaging. And you can see that they perfectly co-localize here. So we do get subnuclear spots and perfect registration among those spots. What I really want to highlight here is when we go to head-to-head competition against other iterative fluorescence imaging, we always win. That's not approved by our legal system, but I can tell you that every time we don't drop fields of view, we have high dynamic ranges. We can see a broad range of intensities. We did a beautiful poster looking at hypoxia at GLUT1, and instead of just on off, we could look at low, medium, high, and we could even divide or cluster by 6 different tiers of expression levels and see different expression patterns. So it's a very robust, sensitive instrument. This is what it looks like. You can see I'm kind of a cosplay here. I like to dress up like my cell dive. It's black with this beautiful red door and name tag there. It has a 2x, a 10x, and a 20x objective in it. It's a very high NA objective for looking through the slide. We image through the slide, so everything is nearly always in focus. We rarely drop a field of view. I mean, this is precious human tissue or other tissues. You don't want to lose fields of view in your imaging. Again, the imager itself can image up to 2 by 4.5 millimeters, and then it's, of course, inside of our processing station is 1.7 centimeters by 3.7. So I'm going to go ahead and pop over and say, what does automation look like for the cell dive? It's paired with this bioassembly bot by Advanced Solutions Life Sciences. They make a number of GMP robots for 3D printing of tissues and organoids and things. So this is a very easy task for them to work with us on. So here's the cell dive on the right. Here's the BAB on the left, as they call her. And I'll show you what that looks like inside. So it can completely process and image 15 slides at a time. So we can have 3 stacks of 5 of these Clickwell assemblies that hold the slides. And you can see there's a little pipetter pipetting into the workstation of slide number 1. This is slide number 2. This is slide number 3. They are barcoded so that they can be tracked when they go into the instrument. It knows that this is Peter's slide number 1, protocol 2, round 3, or Charlie's slide number 7, round 4. Let's go longer. Round 30, running protocol number 10. So they can do them all the same in a big batch, or you could have independent slides running asynchronous from each other. Okay, how expensive is it to run? We use complicated chemicals. We have PBS. Okay, we have glycerol. Okay, so these are very simple chemicals. And then here is where we keep the cooled station for all of your antibody cocktails. And we can hold up to 15 different rounds in there for 1 slide, or one round for 15 slides. As you can see, by creating this assembly line, we're going to continuously feed the imager. The thing with doing this work in bulk when you're doing batches is staining can be done in the same time or near similar time, and that does not scale linearly, right? We're wanting this to go fast. What scales linearly is imaging. So we want to keep the imager with 100% duty cycle, and that's why we're processing these slides off instrument. If you're looking to...
Antibodies can be multiplex together? Like if you have like 60, you have to repeat this process 60 times.
So we do 4 colors plus DAPI at a time, because DAPI is, so if you're doing 60 markers, that's going to be 15 rounds, right? Does that answer the question? Okay. Okay, so that's just a touch base. I realized that I was going to go a mile wide and an inch deep here. So what about 3D? So we have Spectraplex. What does Spectraplex allow us to do? Here we have a particular, okay, I thought I was going to play, there we go. So this is actually 70 micron thick section with 15 different immune oncology markers, and it allows for a very high depth of information to be gleaned from just the structure of this tumor. And with 15, it's not really discovery, but you can really map out where things are, what they are, and what they're doing to a really nice level. And with this number of markers, we're able to ask questions about, or at least identify lymphoid B and T cells. We can ask and see where are the myeloid cells. We can look for lymphoid cells fighting tumor growth. We can look for another cancer stromal and extracellular matrix markers, perhaps looking for CAFs and epithelial and endothelial cells. So highly multiplex image analysis is functional for description of the cancer tissue with these 15 markers. So this is in 3D, but you may think, I don't need 3D, but what if you wanted to look at organelles? You may, then you would want to have the resolution that you can control with a confocal, or depending on the skill set of your frozen sectioner, the wide field-based systems do really well at 3 to 5 microns. When you're working with a new postdoc, they're usually doing 10 microns thick. And so the ability to occlude the out of focus light, you'll get much higher, better segmentation on the data if you're using a confocal of those somewhat thicker specimens. So it really depends on what your goals are for which technology you might want to choose. How do we do this? Well, what's really cool with the Spectroplex is we're able to actually create this ability to define your panel by, you can't really see it here, but we have a variety of markers. We have, or these are floors, these are markers. We create a virtual conjugation. That's our virtual fridge. We put those over here and it defines exactly how it's going to collect the data. So we define our panel. Now here's kind of the secret sauce, is we have a number of markers that are overlapping, but it allows us to create, it defines what our group stain should be as a control. Then it builds this unmixing matrix so that when you run your 15-plex, you get perfect segmentation of all of those different signals. Once that's done, you can now run multiple samples through the system and get on-the-fly spectral unmixing of 15-plex. That's the software. And it's only really enabled because of the hardware that underlies it, where we're able to have complete spectral control over our emission from 400 to 850 nanometers, and complete control of our spectral excitation with a white light laser that goes from 405 to 790. So it's the ability to kind of have our base confocal technology we've already had, and then layer on this almost hand-holding technology, I would call it, on how to do your Spectroplex imaging to do this unmixing. Okay. I apologize. I have very few slides on this because I was trying to, okay, I'm from St. Louis, Missouri. It's where I moved to. And we like to quote Mark Twain. And he always said I would have written a shorter letter if I'd had more time. And I took extra time and shortened this. So I've removed some of the other data on here. So AI visualization and image analysis. So I really kind of want to bring your brain in. We hear AI all the time, and AI is basically trying to teach things to software to think like we do, right? But there's kind of two different tiers on it. We're going to have one that's more like how you train a technician, or, well, a new technician. An experienced technician is going to run with it. A new technician or an undergrad or something like that, you train them on this is what you want to look for. We'd call that machine learning. Right? And so we have machine learning tools within Avia. Now, you may want a subject matter expert that really knows things just when they see it, because they've seen so many. And that's actually deep learning or deep neural networks. And we have that in there too. So we can use this to really view and interact with the data. We use deep neural networks for identifying our cells using both nuclei and multiple membrane markers so that we get very robust segmentation of cells in a very dense heterogeneous tissue. And that's not easy to do, but it's very robust with Avia. And then we can go into identifying phenotypes. Maybe you want to train it to tell it what you're looking for. So we can use machine learning or phenotyping within that. Phenotyper, I think, is the term for that one. Alternatively, we can actually do unsupervised if you wanted to kind of tell you what's there kind of just by the biomarkers using k-means or phenograph Leiden. For identifying those phenotypes. Now, once you've identified all of these, you probably want to go in and generate some insights with a variety of other graphical interfaces. So just to let you know, we can take our multiplex 2D or 3D data input. We can create our very robust segmentation. From there, we can classify the data. We can look at what's our confidence level for confidence level mapping and QC of the data, moving into a variety of data exploration tools like violin plots, dimensionality reductions of UMaps, TISNEs, PAC maps. Did I miss any? No, I got them all? Okay, thank you. Or we can go into scatter plots or bin scatter plots, Pearson's correlation heat maps. Now, what's kind of cool about this is maybe you're machine learning, you don't want to click on, okay, here's my CD45 cell. Maybe you actually want to go ahead and look at your plots because you're used to doing a lot of flow cytometry and you want to go by your binning. You can actually just draw regions around those areas of the plots and it would immediately map it. You could use that for training your different models. So it's a very robust platform that lets you generate a lot of insights from this data. Now, I want to highlight something else that's really unique. I mentioned that at least with CellDive and SpectraPlex, we're able to look at a broad range of signals. So we're not just going to look on, we're not just going to phenotype on plus minus, we're going to phenotype on level of expression. But with Avia, we're not going to just go on intensities. We can actually phenotype on morphology. So we can really use this in delving into different forms of neural tissues, which are probably collected on the confocal, so we can actually trace these rather convoluted things and then start looking for where are the activated glial cells and things relative to different amyloid plaques. So that's all I covered on analysis. So now that we have all these shapes of these tissues, I may want to look at other types of analytes or go deeper into protein where we don't have antibodies for them. We could. So now I'm going to go into the LMD, laser microdissection. And what it allows us to do is get pure contact-free isolates that we can use for our downstream analysis. And because it has a lot of automation, it can be set up for very high throughput capabilities. And the basic workflow is, of course, you take your slide, you treat it, you image it, you cut out what you want, you do your analyte detection, and then you do your quantification. This can all occur with high precision because we don't move the stage to cut the sample. We actually direct the laser. And this allows very precise steering of the laser to cut out very specific areas. Now, I mentioned what we really want is very pure samples that get high signal to noise that aren't contaminated by other things. Our method of collecting the sample is to collect it by gravity. So we're not like using anything sticky that would pull off cells in addition to our regions of interest. So this is a way to get very precise cutting and very clean data. And this can all be automated to put them into multi-well plates. So we can really start getting some wonderful throughput. The types of things we look at in our isolates could be looking at things for actually isolating nuclei and sequencing their DNA. We could move into ways of looking at RNA for single cell seq of different regions. We could also look at collecting a whole bunch of plaques throughout the brain to see what's in the different tissues or individual regions for analysis. But what I want to highlight is kind of a new one that's kind of come out of Matthias Mann's lab at Max Planck, and that's deep visual proteomics using this technique. And so the basic flow here is, here's this beautiful tissue section. Perhaps it was from BioChain. We want to go ahead and process that tissue and to get our signal cells identified and the regions of interest identified, and then cut them out in the laser microdissection and go into mass spec. And from mass spec, do our bioinformatics analysis. A specific one here is really a very heartwarming, exciting story. So a number of patients will respond adversely to different drugs, and you can end up with a lot of skin disorders because of this poor drug response. And so here's a number of them. The one we're going to focus on here, even though there's a few of them in this paper, is this toxic epithelial necrolysis. And patients will actually start to shed their skin because of an inflammatory response. It's 30% fatal. And so this is really a problem and we really don't understand all the pathways. So what they did is they went ahead, and if we look at the process, they took the patients, they did multiplexed immunofluorescence. I think I might know a company that provides such things. And then they were analyzing that data. Can anyone give me a name of a software that might do that? Oh, come on. Avia, there we go. And then they went ahead and they cut out those sections with an LMD. You can see that, and then they did analysis of those different proteins. In here, they were able to, from this immunofluorescence, separate keratinocytes from immune cells and then do proteomic analysis on the different populations. There's a lot of data. I'm not going to go in here, but in this volcano map over here, they found a lot of inflammatory responses that were instilled in this process, including JAK and STAT. So they decided to look more closely at the JAK STAT with immunofluorescence, and they could see this particular, there's healthy, 2 different diseases I'm not going to talk about in 10, where it seemed to be highly upregulated here, in addition to these other analyses. And so they're focusing on this 10 disease and the JAK STAT pathway. I don't have the slides up here, but they went ahead and they tested this on tissue culture models and validated that the JAK pathway was activated, and inactivating it could reduce some of those pathways. They did it in a mouse model, and they could show that they could alleviate it. But what's more exciting here is, I should have warned you before I put this up, but this is a patient. They treated 7 patients, and all of them responded getting re-epithelialization in 30 days. So kind of to just do a quick little review, they used deep visual proteomics of archived tissue biopsies, and they were able to get 5,000 proteins from keratinocytes and skin infiltrating immune cells. And they really noticed a big influx of type 1 and type 2 interferon signatures in the immune cell and keratinocytes, and a lot of phosphorylated STAT1. And I think what's most crucial here is to come down here and realize that treatment with JAK inhibitors was safe and associated with rapid cutaneous re-epithelialization and recovery from 7 patients with 10 disease. And I think, I could be mistaken, but I thought this all occurred within about a 6-month period, which is amazing to go from beginning to end. So I think it's a really wonderful story. I'm going to circle back to CellDive for a bit. I know it was, they said everyone loves to say their instrument is the best and can image well, but seeing is believing. And we do have a proof of concept program for people wanting to evaluate instruments. We have a number of panels for you to choose from human IO to mouse IO to human neural markers. And I had listed in my abstract, I would talk about neural diseases, but my time didn't allow it. So go ahead here. I have 2 QR codes. Please scan them with your phones, and you'll access the posters, which we look at the tissue we got from BioChain on both healthy and Alzheimer's brain. And we're able to do both some really good spatial imaging and spatial analysis on those tissues. So thank you. Any questions?
Was a lottery.
Yeah, I should have picked
Good.
one and gone deep, but I decided to go broad.
Right, yeah.
I
So
should
what-
stay here.
How long does it take for for like 50 plants? How long?
Oh, that's an excellent question. I always hate that question too, because what the power of CellDive is the ability to do multiple slides at a time. So it's really going to depend, different markers at different exposure times. So I'll just give you a couple of metrics for you. So if I was going to tell you one slide, so if I'm doing a size about my thumbnail, because that's kind of one of the sizes out there, about a square centimeter, one round would take about 3.7 hours of one slide. But if I was going to do 10 slides, it would only take 8 hours. So I could get 10 slides in the same amount of time. So it's not like you're just putting 2 slides or 1 slide on there and waiting all this time till it's done. We can actually put on multiple slides. Slides can go on at different times with each other as they go through these processes. So it really,
And how big
it's
is
really
that
very-
region?
oh, I left my backpack back there. Come by my table later. I've got the thing. It's 17 by 37 millimeters.
Okay.
So 6.29 square centimeters.
So you can put a ton of tissues on
A
it.
ton of tissue.
Okay.
Now, you have a ton of tissue. It's going to take a lot more time than a
Time,
little
yeah.
tissue because it's field of view, right? However, it's the same amount of antibody, right? So you have increased time, but reduced antibody per tissue section. So, right, everything's cost benefit analysis.
So this is mostly discovery, right? Like most of these technologies. So you have targeted like small.
You know, it's really interesting. There's like to be spec wars out there about how many do you see, and I do have people that are doing 150 plus markers, but the bulk are probably doing 20 to 40 markers, and it's usually I've got another paper that I'd hope to talk about, but I originally was thinking I had about 45 minutes, but I didn't. But there was a beautiful paper where they only used about 16 markers, one for just differentiating the tissue and the other one looking at different immune cells and the cancer cells. And they were, this was retrospective study looking at chemotherapy. Now I'm drawing a blank on which cancer it was, but they were able to then track and look at the specific cells, and that was the PD-1 minus ones that were responsive and had a good patient survival rate, whereas the other ones didn't. And this was not a huge number of markers, right? So it really, if you have your hypothesis in mind, you can really drill down and just do, a smaller number of markers. But if you're really in discovery trying to figure out that's possible too.
Thank you. We can take a short break, like a 10-minute break. Thank you.
Thank you for tolerating the fire hose.
Thank you, Rick, for a fascinating and a brilliant fusion of science and imaging presentation on spatial biology and the power of omics in spatial context. Your insights into multiplex spatial data generation in 2D and 3D, the vignettes of cancer and neural pathologies offered a compelling look on how imaging and data analysis are transforming our understanding on tissue biology. We truly appreciate your time and expertise. Thank you once again. I'm sure with the first set of presentations done, we have had an insightful ideas and offered meaningful takeaways from all of their research. So it's time for a dose of coffee. So we'll be having a short 15-minute coffee break. Make sure to network, grab a cup of coffee, and if you haven't yet, double check that you have entered into our raffle. We'll reconvene here at around 3:30 p.m. sharp to continue with 2 more exciting presentations. The coffee is right here at the back. We'll see you soon, everyone. And welcome back, everyone. If the coffee didn't wake you up, the next session definitely will. We are starting our afternoon session with a bang. Our next presenter is Xiaoshan Wang of Spatomics, who will introduce us to their next generation multiplex assays. With over 15 years of biotechnology and clinical diagnostics, Xiaoshan brings a wealth of experience to the spatial field. Before his tenure at Spatomics, he held pivotal roles at Biotechne and Takara Bio USA, focusing on in vitro diagnostic and companion diagnostic product strategies. He earned his MS in biomedical sciences from Virginia Tech and an MBA from UC Berkeley Haas School of Business. Today, he will be presenting on the topic multiplex spatial profiling with cleavable TSA fluorophores. The floor is all yours, Shashank.
Okay, thank you. I want to make another announcement that before I move on, I actually worked at BioChain for 13 years. That's longer than some of the BioChain colleagues over here. So if you've been using the BioChain tissue for like over 10 years, if you have ever come across any lot number starting from A or B, that could be my process. All right, so after I left ACD, I joined a startup called Spatomics. So I do see that there's a lot of pinpoint which is not resolved right now. So at Spatomics, so we're small. We're only 2 years old, but we are not for sure we're not as well known as a 10x or a Leica. So they don't need to introduce themselves, but I do, because since Spatomics is only 2 years old. We founded 2 years ago. So we are headquartered in Connecticut, then very close to Yale. And what we do here is, although the company's young, but we're doing a very innovative technology that give you a cleavable TSA assay. So traditionally TSA is not a cleavable. So I think there are, no matter it's the genomics or like they've been mentioning a lot about spatial biology. Why we're doing spatial biology? Because kind of a Nature Method of the Year has already endorsed it. It's 2020, the transcriptomics, and also 2024 proteomics. So there's a highly demanded in the market, and also they're not mature. Market is big and it's not mature. So there's still a lot of room for the technology to grow, and also there's still room for new players to play in this field. So that's basically the rationale why we are in this technology, in this field. Let me bring the leader pointer out. Here's some on-market technology that can do a relatively low plex, like a 6 to 12 plex immunofluorescent assay. So one of the most famous ones being the Akoya's Opal assay. So the TSA technology is not new. It was developed like more than 20 years ago by the Perkin-Elmer scientist, and luckily the patents expire, so everybody can do that. So with Opal, the downside is that you can only do probably up to 8. And you have to have a multispectral imager or scanner in order to separate each of the different channels. And 8 to a lot of users is already good enough. But nowadays, the people are using more and more markers on immuno-oncology to tissue samples. So 8 is not enough anymore. And also, with Akoya Opal, one of the benefits is that you can use off-the-shelf antibody. Standard antibody, no modification needed. Well, there are some other technologies from, for example, from a cell signaling to signal star, or from AutoView's in situ plex. They could allow you to also do like a 8 to 6 to 12 or 8 to 12 plex, and without the spectral unmixing. So you can do 4 markers at a time and do a multiple round, like a 2 to 3 round, in order to get the 8 to 12 plex. But the downside is, you can see very obviously, they have to use a conjugated antibody. So once the antibody is conjugated with the oligos are relatively big molecule. Once it's conjugated to the antibody, they may change the nature of the antibody. The antibody may work, but after conjugation, there's a high chance that the antibody will change its nature and may not work as before. So what we do here, we leverage TSA for signal amplification. In the first 2 steps, is essentially very similar to Akoya's Opal assay. We have a TSA, we have a ceramide fluorophore, and we use the traditional primary, secondary, and indirect way to amplify the signal. And the primary antibody is just a regular, off-the-shelf, standard antibody. No modification needed. And secondary antibody, it's the HRP. Normally we use a polymer HRP secondary antibody. And the secret sauce actually here. So we design a cleavable linker in between the ceramide and the fluorophore. So you know that with the traditional Opal assay, once the ceramide fluorophore is activated, it will deposit on or around the target of interest. And then they will form a carbon covalent bonding. This is very, very strong. Normally, it cannot be break. So what we do is that the cleavable linker we have can be easily opened with a mild cleavage region. So you can release the fluorophore, the ceramide part is left behind, and then you can redo the staining because the fluorophore again is gone and the HRP is deactivated. I know that the people may be a little bit worried about the stereo hindering or umbrella effect. Well, what we did in our lab is that we have been using the same antibody and detect the same marker repeatedly again and again. We detect it, cleave off of the fluorophore, strip off the antibody, then redo the staining using the same antibody, same fluorophore for 12 times. And then we got a really close, very, very comparable result. So with if you go super high, I don't know, maybe you go 300 or 400 plex, you may run into an issue. But for now, when you're running like a below 100 plex, I don't really see that kind of issue happening. And also, one of the benefits is that you don't need to invest in the new box. So if you're using, if you're already using like a bound, or you're using the BioChain's OncoPrex, you can directly take advantage of our assay. You can use a standard tissue slice, no special slice needed, and then put it onto the auto stainer. And the auto stainer with the OncoPrex, it's I believe the throughput is 36 slides,
and with the Leica bound is about 30 slides. And with our fluorophore, you should be able to process around like a 12-plex slides within 2 days. And the workflow is very similar to the traditional RF workflow. You just deposit the fluorophore and take the imaging. And if you don't have the spectral, like a multispectral imager, it's fine. If you only just have a microscope-based one with a 2 to 3 channel, you can still do the cycling. You can still do cycling. No worried about that. So in a very extreme case in our lab, not in a co-founder's lab, he been using just one fluorescent channel and do over 20 plex, just repeatedly using the one fluorophore. So for this one, you can see that we can also work with molecular instruments HCR Pro. So you could do because molecular instrument HCR assay is a protease-free assay, you can easily combine your RNA ISH assay with your protein assay for a protein RNA co-detection. So after you deposit all the fluorophores and then do the imaging, you can cleave off the fluorophores using a one-step mild cleavage reagent, and then repeat it the staining cycle until the desired plexing levels are reached. You can then using either the commercial software like Indica Labs Halo or VisualFarm, or you can use the like the open source software like ImageJ or QPass for image analysis. Now we have 12 cleavable fluorophores. Majority of them are 1-to-1 equivalent to Akoya's Opal assay. And I feel very lucky to see everybody here because Akoya also have a event today. So I want to show you guys something a little bit different from the Akoya's Opal assay. So with this 12 fluorophores, you don't need to use all 12 together. If you have a imager that can have the capability to do 12 together, by all means do it. But if you only have a imager can do 2 to 3 or 3 to 4 color or up to 6 color, you can cherry pick whatever fluorophores working for your imaging system and then do it in a multiplex way. In the following, I want to show you guys some of the example we have done. So here is the tonsil tissue. Of course, the tissue is coming from BioChain. So we feel very lucky to have a BioChain as a collaborator because BioChain has a wider range of tissue choices, and also they're garbage in, garbage out. We got a really high quality of a tissue for our experiment. With this one as the tonsil tissue, then we did a 2-round staining using a manual way. So the first round we did 5 flags with a DAPI and also with a CD4, CD8, FOXP3, CD20, and PanCK. So with Akoya's Opal assay, you probably know that you have to follow a strict staining order. Normally, it's from a low abundance to a high abundance, and they generally follow a very strict rule to using what fluorophore for what marker. But for our assay, since the assay is cleavable, so you have a lot of flexibility. Or if you are making any errors, human errors, or if your antibody didn't work, you could just strip them off, cleave off the fluorophore, and then repeat it again. So for tonsil tissue, maybe it's not that precious. But if you have some patient sample or you have some rare tissue, you could you have a more so our assay is more error tolerance. So you could have a second chance of staining. Well, this is after the tissue imaging of the first round, and then we apply this cleavage reagent, and you can see that majority for this one is taking from the Zeiss Zen software. You can see there's a different intensity of a fluorophores. And after the cleavage, pretty much everything goes back to the baseline, and your DAPI channels, the signal stand up, and you pretty much don't see anything after the signal cleavage. And at the same time, we are able to remove more than 99% of the fluorophores. So after the first round, we did another round of staining with the 6 markers. And here's you can see that some of the markers still have a relatively high abundance, and we are able to still image them really well without the carryover from the first round. This is a recent publication from our co-founders lab in Arizona State University. So this is his graduate student that published a paper with a manual staining with 38 flags. We don't really want to do that. 38 flags is a really long time, but for students as a publication, I think it's totally fine. But for us, we want to do it probably using at least using an auto stainer. Now probably the BioChain's Uncle Pro X or the Lycabal. So with this one, he did a 36 plex of protein, and he chosen the 500,000 cells for their like expression level analysis, and that they are able to cluster them into, I believe, the 12 clusters, and they can apply those clusters back to the tissue, to the whole tissue, and to figure out where these clusters are located. Here's also a manual assay from one of our collaborators in China. So they have done a non-plex on the FFPE mouse brain, and this collaborator actually also an antibody manufacturer. So they are using their own antibodies, because we are using off-the-shelf antibodies. The compatibility is really well, and we can see that the image looks really beautiful on the mouse brain. As I mentioned that nowadays, more and more users they want to see both the transcriptomics data and also proteomics data. So our assay, they offer a relatively low plex, but I believe that with a 10x, no matter the 10x system or the Leica system, they could be a very good complementary. They could go upstream or downstream to their high throughput and a high plexing system. So with this one, you are able to do 102 RNA markers using molecular instruments HCR Pro. So here we can see that this is using the UBC. UBC is a very highly expressed marker, and of course you can see a lot of transcripts. Each of the dots representing one mRNA. And for this one, is a TBP. TBP on HeLa cell, on average, the transcript per cell is only 4 or 5 copies. With a lot of transcriptomics platform, probably including RNAscope's HiPlex, they're not able to really see so many of the transcripts. But with the molecular instruments, HCR Pro and RNAscope's multiplex V2, you are able to see very low copy number of a TBP from a HeLa cell. And on the right side is the molecular instruments HCR Pro. They're newly launched a 2-plex assay. Basically, we're staining one of the one, which is a long non-coding RNA. The other one is PPIB, which is a very commonly used housekeeping gene. So you can use this housekeeping gene, and I believe that BioChain also have this product, which is HCR validated tissue, which you can use the PPIB to assess the RNA quality in FFP tissue. And afterwards, here's an example from the UBC staining. We applied our cleavage reagent to the sample, to the stain sample, and afterwards you can see that you got a really clean result with all the RNA signals removed. The next one is the automated RNA, the protein code detection, standing on the BioChain's Uncle Pro X system. It's really easy to use. And we have our instrument installed about 3 months ago, and we've been using that instrument since then. And the only downside is that we are using a NAS server to store our data. But once we had the auto stainer, and the NAS server got maxed out really quickly, because we are able to generate so much more data than comparing to the manual assay. So this one this is a PPIB plus 3 protein, which is 3 subspecies of a T cell, CD4, CD8, FOXP3. You can see that those are 3 subspecies that have a really good agreement. So there's no co-expression. And if I can, let's see if I can zoom in. Oh, yes, great. So this is a normal brain. And within the normal brain, you don't really see that many of the immune cells, but the PPIB, this is the yellow dot, that still stand out really good. And on the right side is the, let's see what it is. It's a breast tumor. Of course, in the tumor region, they have a lot of PPIB expression, and also expected within this tumor region, you will see a lot of different immune cells. Okay. This is one of our recent work. We did like a 6-round staining, with the first round being the RNA ISH using HCR Pro for PPIB, and downstream, kind of a plug and play. We did a 5-round of a protein marker, each round with 3 to 4 markers. And this is by all means, this is not the best data we have. Our assay, we're a startup company. Our assay is not as mature as the 10x genomics assay or the Leica microsystems assay. But since we're small, we're very nimble. We are making improvement almost every day. And I believe that in no time, we'll get the assay looking even better. And also plexing level wise, it will even higher. And also, I remember hearing Rick mention that there's a lectin detection. Yeah, so we are also working with vector laboratories for the lectin and protein co-detection. So with lectin, it's a glycan binding protein. So they can be used similarly like an antibody to detect very unique glycans. So some of the glycans will change, will alter in the Alzheimer patients samples. So based on the glycan expression level and combining with RNA with the protein and the potential RNA expression, you can see a lot more. You can gather a lot more data from the same piece of the tissue. Here, I think this one Vidya also mentioned in her deck. So this is a pilot study. I really appreciate the support from BioChain that we are able to get our hands onto some of the post-xenium slides. So I know that with the 10x is inline protein detection, you get a lot of ease of mind to get just one more day. You get so many protein markers detected on the same tissue. But since that one is not a kind of an open panel, if you have some custom antibodies or protein markers you really want to see, you probably can try this assay. So with our assay, we're able to detect like 9 or 10, I cannot recall exactly, but we're detecting 9 or 10 protein markers using our TSA-based assay on the post-xenium slides. So I want to wrap it up really quick with our assay. So you could leverage your existing instrument or even do it manually. No matter you do it on the auto stainer or doing it manually, the assay workflow is very similar to existing Opal assay or the traditional IHC assay. And afterwards, you could image your slides all in one shot with the high throughput scanner. For example, like the top one is Olympus's VS200, or you could use also the Pheno Imager HT, or I know a lot of people still want to call it Vector Polaris. And we are working very closely with Zeiss, so we are also imaging it on the exoscan. So I think with this setup, you are able to pump through a lot of slides with a relatively lower plexing level and with a much higher throughput. With that, I'll wrap up I think I did it pretty quick. So, so if you have any questions, I'm happy to answer them. Thank you. Sure, yeah.
Just a question about your tyramide non-umbrella effect. So it's kind of like, I always get questions about steric hindrance of just the antibodies, and it's always written in the articles that it's a problem, but
yeah,
none of my people see it, and I've not seen it.
Right.
As far as just antibodies, even with drug conjugates. So is there something different about your deposition compared to, say, an Opal, or why do you think
well,
there's no umbrella effect? Or
theory-wise, this is the same as an Opal assay. A lot of people have been talking about and asking us about the umbrella effect. So we have done 2 experiments. One I mentioned during the presentation, that we've been using the same antibody, repeatedly staining the same marker, and we are getting a similar reading each of the cycle. And the second experiment is kind of also an interesting one. So we stain CD3, 2 different isotypes of CD3. I think a CD3D and a CD3E. And then we stain the CD3D and H3. So they are very CD3D and H3 are very further apart. One is a surface protein, one is a nuclear protein. And the CD3D and E, they're supposed to be two epitopes very close together. And with this experiment, we are getting similar CD3D reading, no matter if it's co-stained with the CD3E or co-stained with H3. So to us, at least at our current the current flexing level, we don't really see too much of the stereo hindrance affecting the results.
Beautiful. You think are they just recommending that the others are they just recommending overdevelopment?
What do you mean over?
Well, if you leave it in, if you let the HRP run longer and longer and longer, you're going to deposit more and more and more and more. So maybe they're overdeveloping the samples, and that's why you get more deposition. Whereas you're creating a more controlled environment for.
Right. That's definitely possible. Yeah. But with our assay, and I think one thing you remind me, you mentioned that if you are using antibody stripping, kind of you will destroy the epitope, for sure. So with the OPAL assay, normally it's recommended to use an antigen retrieval to get rid of the antibody. So we could do the same thing using an antigen retrieval to get rid of the antibody, to strip the antibody, but we're also using a low temperature stripping buffer, which worked at 40 degrees C or 50 degrees C. They worked equally well, if not better than antibody retrieval, antigen retrieval. And they will also help preserve the tissue better. So currently we have, we're working very closely with the BioCare. They're being very supportive, and we have our assay automated on their OncoPRX. Also, we have a automated protocol on Leica BonRx. And also we are working closely with some users trying to automate this assay onto a Discovery Ultra. Yeah.
Very good.
All right. Okay. All right. Thank you, everybody.
Thank you so much, Shaoshan, for technically rich presentation in multiplex spatial profiling. Your deep dive into cleavable TSA fluorophores, pyramid signal amplification, and RNA protein co-detection highlighted the exciting capabilities of spatomics. We are almost at the end of our program today. To wrap up our technical sessions, last but not the least, please welcome Darius Fuzair, SVP Commercial at Singular Genomics, presenting on the topic High Throughput In Situ Multi-omic Profiling of FFPE Tissues. Darius brings a wealth of experience in genomics and product innovation, leading the charge at Singular Genomics to develop cutting-edge sequencing technologies. Please welcome him as he showcases the G4X spatial sequences capabilities for FFPE tissues and integrated single-cell multi-omic analysis. Here you go, Darius.
Well, thank you very much for the kind introduction, and thanks to the BioChain team for hosting us all today. So I might be telling you about a new product that's in development. That's a new spatial technology called the G4X. This is a very high throughput in situ multi-omics platform. So Singular is based in San Diego. We've been around since 2016. We've been in development of the technology since that point. In 2019, we really started to do in situ sequencing on our underlying sequencing platform. So what really drives us is I think we have a collective understanding of spatial biology will play a very important role in precision medicine moving forward. It's a rapidly growing field. It's increasingly clinical. We really view this as moving into routine pathology. And right now, primarily, it's just constrained by very high prices and very low throughput. So that I think restricts the ability to move into translational and clinical. So today's studies are typically small just because they're expensive and low throughput. They take a long time. But I think tomorrow, especially as we look at larger retrospective cohorts, these are much larger studies with higher power across heterogeneous patient populations. And then we also see I think Rick nicely summarized the push into 3D. And one way to do this is 3D reconstruction of serial sections. So you can analyze tissue in 3D with more sections. And really, together, this could be an exponential issue where studies of tomorrow, across more sections, across more samples. So this is really at the heart of what we are thinking about at Singular and what's really driven the innovation over the last couple of years. So everything at Singular is done in-house, starting from the nucleotide chemistry and protein engineering, on the sequencing itself, all the way through, systems integration, optical, electrical, mechanical engineering, to bring forward a platform that's capable of imaging over 2 billion pixels per second, and across 40 centimeters squared. So we've actually been in the market for a couple years with our sequencing platform. And we've got a couple dozen sites that have been running this for about 3 years or so. And last year at AGBT, what we announced was our ability to move forward the innovations that we've done in situ. We always had a second platform in development. We were able to move those onto our existing sequencing platform. So what that is called the G4X. So this is it's high throughput in situ multi-omics. And by high throughput, we're talking about 10 times the throughput. We've about 40 centimeters squared that we can process per run, and about 128 samples per run that we can do. What that allows us to do, importantly, is bring down the cost of spatial profiling from multiple thousands of dollars to hundreds of dollars, which we think is important for moving the whole field forward. And the performance of the system, so it's subcellular resolution, 0.5 micron resolution, and that gives us precise single cell insights. And then we're integrating transcriptomics, proteomics, and fluorescent H&E all in the same section, all in the same FFP section. And then lastly, in development, we have a novel capability called direct sequencing. So it's a fourth modality where we can actually gap fill sequence variable regions of information within that sample as well. So these are the readout modalities. Starting from the bottom, we do a H&E. That's a fluorescent H&E. So eosin's already nicely fluorescent, but we do a fluorescent version of hematoxylin. This is nice. It allows you to see the tissue morphology, connect into standard histology, maybe digital pathology data sets. We also do a 15-plex proteomics panel, and we do a 350-plex transcriptomics, or 350 RNA genes. And that's a barcode-free approach, which carries some unique advantages as well. And then that last modality is called direct sequencing. So this is where we can look at things like CDR3 sequence, where we can sequence the up to 100 bases in situ. We can look at cancer mutations, gene editing, and things like that. So the workflow itself, we've developed a patented high throughput tissue transfer mechanism that allows you to go from just a simple section off the block into a really nice array onto the flow cells on the slides. We've streamlined the sample preparation. It's about a 2.5-day prep. And then we have a panel designer so you can customize both RNA and protein. The run itself we have very high throughput, but it's a flexible use of that throughput. That's also really nice. I mentioned the system's actually been out in the field for about 3 years, so we've gotten a lot of feedback from users in terms of the system's performance, how we can improve that. We've made a lot of improvements, so now it's a very field mature underlying platform with high reliability and robustness. And it's nicely intuitive in terms of starting the system, loading it, operating it, so you can really run it in production mode. And then the analysis, we've got we output standard single cell and spatial biology file output. So everything's done at a single cell level. We do onboard cell segmentation, produce a transcript localization XYZ coordinates. And then you can plug our output into any open source pipelines. We also offer a web-based visualization tool as a nice way to navigate the tissue and see that the multi-omic information in context. So working with tissue, basically what we've developed or the team's developed is a mechanism for going from an FFP block into a very high throughput slide format. So you take your section, you'll take your section off the block, float it in a water bath, and we actually provide these hydrogel pads. You float the section onto a pad, and you don't have to be precise about it because you're going to come in with this tissue punch tool. We have blades of two different sizes, a 4.5 by 4.5 millimeter and a 10 by 10 millimeter blade. That goes in the punch tool. You punch through the tissue and hydrogel construct, your ROI, and then you deposit it on a tissue transfer array that we have. So this is a tissue transfer device. Once all your tissues are on that, then it's just you close the top and then you put it on a heat block, so heat and pressure, and you're left with all your tissues nicely arrayed on the slide itself. It's very simple to do. You know, you can do a full run of up to 128 samples. If they're serial sections, you can do it in about 30 minutes. If they're all different samples, then probably about a half-day effort. But it's nice because it's very gentle on the tissues, very scalable to do. You always are maximizing your imaging area on the system. And you don't need to score the block, create TMAs. We see a lot of larger studies that we're working on TMAs, moving into this kind of 4.5 by 4.5 millimeter format. And it's actually nice because you can store at different points. So you can actually store the section on the hydrogel pad for about 4 weeks, which is nice because you could have one lab section, and then you could send that section on the pad to another lab that actually has a system. Or you can transfer on the slide, and then in that case, you can have the lab transfer to the slide. In that case, you can store it for about 6 months or so, or 4 to 6 months. So it's kind of flexible in sectioning when you want to, running when you want to. Once you're through the tissue transfer process, we have a novel in situ chemistry that the team's developed. Basically what this is we do rolling circle amplification on padlock probes. So for transcript counting, what we're doing is we're targeting 3 to 12 probes per gene. We do rolling circle amplification, and then we're actually directly sequencing that foot binding region, which is nice because then we actually have probe level specificity. For direct sequencing, we actually sequence between those foot binding regions, which is nice. That's how you can get 100 bases of variable sequence information. And then for protein detection, these are oligoconjugated antibodies. We do rolling circle amplification, and then we actually image a single base. We have a 4-color chemistry, so we can image 4 proteins at a time. We do multiple rounds of that. We also do multiple rounds of the transcript sequencing as well. And what's nice about this is we can basically sequence 1 round of transcripts, terminate, go on, sequence the next round of transcripts. So what you could actually have 2 transcripts that are directly on top of each other, but since you're not imaging them at the same time, then you can optically deconvolute those. So it's really nice. Well, we have 0.5 micron resolution and maybe a 300 nanometer pixel effective pixel size. We can actually optically deconvolute even beyond that, since they're not detected at the same time. So we've seen really great performance across very degraded FFP samples because we have very small foot binding regions that we need to actually get the sequence information from. Once you're, this whole process, like I said, is about a 2.5 day wet lab workflow. And then once you're ready to load the system, that's where you'll put in the actual flow cells. So we have 2 configurations. We have a 4-lane flow cell, and you can get 32 of those 4.5 by 4.5 millimeter ROIs in there in each flow cell, so 128 per run. And then we have a 2-lane configuration where you can get 10 by 10 millimeter samples in there, so 40 samples per run. And these are actually the lanes are fluidically independent. So with the 4-lane version, you can run a different panel in each lane, so you can run 4 panels per flow cell, up to 16 unique panels per run. And the 2-lane is, of course, 2 lanes. So it's actually even really nice for us internally in R&D because, with each system, each day you can test 16 different conditions because you have 16 fluidically independent lanes. And we have a fleet of these, so we've been able to iterate very fast on the actual platform itself. We also have integrated single cell analysis and spatial viewer. So we take the output files from the system. And so like I mentioned, we do onboard cell segmentation. You get single cell information out of the platform, as well as your TIFF files or images of proteins in the H&E. And these can be ported directly into our viewer, and it's really nice. You can kind of navigate the tissue. You can look at the different channels of proteins. You can look at the transcript layer, the cell segmentation, the H&E, and you can kind of look at all three modalities together. You can, of course, plug those files into any downstream analysis platform. We do have some tertiary analysis pipelines we've stood up internally that we use for service projects. But off the system, you do get a lot of the important QC metrics. You do get images of like transcript density, number of transcripts per cell, all the metrics that you'd really care about seeing initially, you do get right out of the system when it's done, prior to any of this. We have developed our panels in a modular way. So we have 4 off-the-shelf panels right now: lung, kidney, colon, and breast cancer. These are all 100 tissue-specific genes, and they all share a base set of 150 immune cell genes and 50 stromal architecture markers. So it's really nice if you want to just customize a new panel. You can just add 100 tissue-specific genes or your select genes on top of that core set. You could easily add 25 to 50 genes on top of any panel that we have, or you can go full custom up to 350 genes. And then similar on the protein side, we've got a 15-plex IO focus panel right now. And you can actually add 2 markers to that. So we have 2 positions open for custom markers. So looking at the performance of the system, this was a study we did earlier this year. Basically, we took 2 4-lane flow cells. So this was 64 samples. We had the 300 gene panels that we did, and we did a different panel in each lane. And we did different sections, so both cancer and normal and duplicate within the lanes, and then we include tonsil controls. One of the nice benefits of having all this space is we can include control samples in every flow cell, but for here we just did one in every lane. And the performance shows very high sensitivity, so we can look at that in 2 ways: number of transcripts per cell and the number of unique genes per cell. And what this is showing both the normal samples in there and the cancer samples. So the ones on the bottom and kind of a different color, those are the normal samples, and then the ones that with higher level of expression, those are going to be the cancer samples with higher transcripts per cell. So you can see we're about between 100 to 200 transcripts per cell for cancer samples, and between 40 and 60 unique genes per cell for a 300-plex panel. So I think very good sensitivity. And then on the specificity side, we were really targeting 1% false discovery rate when we were developing the platform. I'm really happy to say the team's brought that down below 0.1%. And we measure that in 2 ways. We look at negative control probes, so probes that are physically in the panel that don't target the human genome, so they target alien sequence, and then negative control sequences, and these help look at decoding errors or sequence-specific errors. For reproducibility, we looked at serial sections in 2 different flow cells, and we look at that both at the probe level and the gene level. So we have exceptional reproducibility on the platform, kind of shown here, and then also just nice separation between our negative control probes and sequences and our targeted probes, again indicating very good specificity of the platform. One thing that's nice about looking at RNA and protein together is you can look at the correlation of the 2. So, transcript markers that like CD3D expect to be correlated with the protein marker CD3. You see good correlation, but not perfect, and that's the point of needing to run both RNA and protein together. But this is really nice for internal, potential validation of protein markers at a single cell level within a sample. And then, like I said, we also have some internal pipelines that we're running that we plug our data through, and we do clustering, single cell phenotyping. You can look at the basically expression plots and mapped onto the tissue, and we have kind of heat plots to kind of show that out, but then also how we're actually identifying those specific clusters by the genes in comparison to the rest and their level of expression. And then we can output the visualizations. So we show the digital H&E, we show the spatial, so the actual single cell phenotypes on the tissue, and then the UMAP, and you're just seeing those different cell phenotypes individually mapped onto the tissue at the bottom. And so this is just an example in colon cancer where we are, we looked at three within that, if you remember that flow cell design. So we had 3 normals, 3 cancer samples, and you can see kind of the single cells identified in the UMAP and the cells at the bottom there, or the cell types at the bottom. What you can see is the very strong visual similarity between the normals, and then a difference between the cancer and the normal patients. So you can see unique cell populations in the cancer that are not present in the normal, and then you can kind of see those cells mapped back onto the tissue on the right-hand side. And then these are just images out of the system, the fluorescent H&E on the left. We have 8 different proteins that we're showing here just for visualization. You can view them all together if you want, just gets a little messy. And then the transcripts. So kind of zooming in at the kind of single cell level, you can see some of the key protein markers and around the structure, and then some of the immune markers as well. And you see really nice morphology and good ring-like structure around the cells as expected. So one fun thing to do is, oh, I think I broke something.
The NAS drive filled up.
Yeah. Big file. I think that was the previous one. Yeah. Maybe it's crashed. Maybe that singular G4X overview. Perfect. Thank you. And let me see if I can quickly go through these here. Oh, hopefully I didn't crash it again. There we go. Let's just get into the fun parts here. So one nice thing about the throughput is we can do 3D reconstruction, at scale. So back at AGBT, we showed that, this is 10 sections of renal cell carcinoma. We put on a single flow cell. These are 5-micron sections, so it ended up being a 50-micron reconstruction. But about 6.2 million cells, 438 million transcripts. And these are the individual sections that you're seeing visualized across. What we can do is when we collapse those down, then we can look at the performance of those in terms of the number of transcripts per cell, meaning unique genes per cell, very consistent across each of those. But we can do 3D cell neighborhood analysis, which is nice because, as Rick was mentioning, biology is inherently 3D. So being able to look at the cells above and below can be potentially important in things like biomarker discovery and just overall analyzing the sample. So we did cell neighborhood analysis in X, Y, and Z dimensions. And all of those kind of cell neighborhoods composite on those 10 slides, the visualization here on the left. And in white, you'll see an ROI that's popped out. So this was a section that actually spanned a tumor normal boundary. So you see normal kidney structures in this ROI. There's cheese wheel. We can see like the proximal tubules, the loop of Henle, glomeruli, things that you expect to be permeating nicely through the Z dimension. We see that. And then in the other section, we're looking at the immune interface with the tumor. So tertiary lymphoid structure, along the tumor and the proximal tubules there. So, I think the nicest thing about the platform is really us bringing down the price. So, we're basically for the larger sections, $750 per sample will be the reagent cost. For multi-omics, $1,000 per sample. And then for the smaller ones, really they're $235 a sample and $320 per sample. So hopefully, enabling large retrospective cohorts, clinical trial analyses to become possible with spatial transcriptomics and spatial multi-omics approaches. So we're in early access right now. We have 4 early access sites up and running the system. We've been running spatial technology services in-house. We've got about 16 successful projects under our belt. And great feedback from the early access sites so far. So really what we're doing is continuing to scale up into more and more partners and institutions through the course of the year. And that's with that, I'm happy to take any questions.
Does your software platform automatically align and create 3D structures, or is that done off?
No, we don't have automatic. I mean, our team has developed automatic alignment, though, I think it works some of the time, but it always seems like there's some manual intervention that's required there. Yeah. I'd say normally, like if you're not doing 3D reconstruction, of course, everything's gonna be nicely registered because we do it in the same sample. So like the RNA, protein, H&E is always gonna come out registered together because it's done within the same sample in the same run. But yeah, the 3D stuff was done separately. Yeah.
Nice.
Yeah.
Does it do Z stacking?
We do multiple Z layers of the image, yeah, of each tissue section. I think 3 to 5. I'm not sure the exact number.
You don't do it for an individual.
Yeah, within the individual section, we do 3 to 5 Z planes. Yeah. Yeah.
You do the H&E staying after you image it?
Yeah, it's on the same run. So a full run takes about, if you run 4 flow cells, fully loaded, it's about 29 hours. And so the H&E is the last piece of that. So we do transcripts and proteins and H&E. And so the H&E is done on board. Yeah.
And automatically?
Yep.
It's a virtual H&E created from DAPI and autofluorescent.
Yeah, yep. ESN, it's a fluorescent version of hematoxylin. Yeah.
Oh, okay.
So it's like a digital version of the H&E. Yeah.
Cool.
Yeah.
Did you say that there's cell segmentation in your software?
Yes.
How accurate is it? Is there any training, or is it just the default?
Great question. Right now we're doing nuclear expansion for cell segmentation. We think it's working pretty well. There's always room for improvement with cell segmentation in general. I think everyone can agree with that. We are working on membrane-based cell segmentation, like membrane and cytoplasmic-based cell segmentation using the protein markers. That's not out yet, but right now we're doing nuclear expansion. Yeah.
With that nuclear expansion, is that proprietary, or are you using something like Mesmer?
I have to ask our software team. Sorry. Yeah.
And how good is the resolution in terms of like the localization of the mRNA if you're showing it in the cell, basically?
Yeah, so 0.5 micron resolution, 300, I think a little bit below 300 nanometer pixel size, but like I mentioned with the transcript detection, we're not necessarily bound by those constraints because we are doing multiple rounds of that. So 2 transcripts can be right on top of each other. So they could, you're not actually bounded by physics in that scenario, right? Yeah.
So you mentioned about you do a transcript copying, but you also do the sequencing.
Yeah.
Are you doing them simultaneously, or these are separate workflow?
So we're actually sequencing to identify the transcripts. So like we're actually sequencing the foot binding region in the transcript. So it's natively in situ sequencing is what we're doing. Now I mentioned the other modality, which is direct sequencing that we call it, or direct seq. That's where we're gap fill sequencing. So instead of just seeing the foot binding region and looking at and counting those, we're actually sequencing between them. So that's how you could see, and I didn't show any of the examples of that today because it's still in development, but we do have a poster if you want to check out the website where we look at, we see CDR3 sequences in B and T cells and tissues, and we can see clonal populations of those immune cells, through the tissue. So very exciting potential application of the technology. I will answer, I think to follow up on your question, I don't know what we're using for cell segmentation, but you can do cell segmentation if you want, if you have your own pipeline that you want to use after the fact. Yeah.
You said that sample preparation is 2.5 days. I guess how long does the process start to finish?
So the tissue transfer process, if there's a bunch of different tissues, it's about a half-day process. The wet lab workflow is about a 2.5-day process. The sequencing is, depending on the number of flow cells, up to 29 hours. And then analysis right now is about 3 days off board. So we provide the high performance compute system with the platform. Those are NVIDIA GPUs. We're accelerating that, but right now it's about 3 days after. You could offload that like we do internally. We have compute clusters, so we can keep the systems running nearly every day. But with the one we provide, it's about 3 days.
So then in total, it's like just shy of a week.
Probably. So you could probably run twice. You can run twice a week with a single tech, if you're planning effectively. And the idea is to get this basically under a day for analysis. So you can run it, potentially 3 times a week, but you're really stretching into the weekend.
Yeah.
And I don't think people want to do that.
Never know.
Great, thank you.
Thank you, Darius, for an excellent and forward-looking presentation. Your insights on G4X workflow overview, advancements in single-cell profiling, and FFPE compatibility are truly promising for the future of spatial biology and FFPE-based research as well. Of course, not to forget, spatial biology is the future of precision medicine. Now, what a fantastic lineup. Thank you to all our presenters today. Raffle winners will be announced later, and our team will follow up with email with all your exclusive discounts. On behalf of BioChain Institute, Inc., I thank you so very much for joining us today. We started this as a flagship space for accessing the latest in spatial innovation and connecting with industry peers who are just as passionate about this work as we are. We hope that we can continue hosting these environments and cultivating these relationships so that the true potential of spatial for clinical research can be realized. Thank you once again to all our amazing presenters, attendees on site and online. Without you, this wouldn't have gone so far very well. Our extended marketing team, Allison and Kimia, for arranging and coordinating so very well with our expert speakers, and also alongside working with my BioChain team, who has taken efforts in organizing this event. And of course, not to forget our valued partners in this spatial journey. Safe travels home, everyone. We look forward to seeing you all achieving next in spatial biology. Have a wonderful evening with our spatial symposium hour. Enjoy tasty bites, refreshing sips, and happy networking. This is Nidhi again signing off from today's event. Thank you once again.